Morning everyone, and welcome to Year 11 Biology with Elite Medics Online. It is 10 a.m. now, so we are going to make a start to the teaching session. I'm just going to check that you guys can everything is ready. Okay, that's fine. My screen's being shared. You guys can all see me and hear me. Okay, so we're going to make a start to the session. It is April. We've seemed to have covered a lot in the previous sessions, and we are heading towards the end of the year now. And I'm sure you guys have been very, very preoccupied revising for exams. I personally feel the topic we're learning about today is a very, very fun one in the sense that we're actually talking about real biology, and I personally find it quite enjoyable to teach, and I'm hoping you guys are going to find it just as enjoyable to learn. As you can see, we're covering a variety of things today, so it may feel like a bit of information overload at times, but we are going to have breaks in between to make sure you guys are taking on the information really well. We're going to be covering infectious diseases today, the immune system, modern medicine, and plant diseases. So, it's a lot of conversation about diseases and how they vary from humans to plants. And as always, we're going to obviously run the session till 3. We're going to have a morning session running from now up until 12:30. Then we'll break for lunch for an hour, and then we'll resume from 1:30 to 3. You guys are obviously very well aware that we're going to run some content, then we will do some interactive questions and answers, and then from there we will then take it on to doing some exam questions just to help you reinforce what you've learned so far. Given the nature of the content we're covering today, I have interspersed a lot of medicine into it as well, just to keep things interesting for you guys, so it doesn't feel very boring and you're not just learning very your specification based things. So we'll make a start. So like I was saying, we're covering infectious diseases, the immune system, modern medicine, and plant diseases today. And we're going to begin today by talking about pathogens. Now, obviously, whenever we start a new topic, you guys are very well aware, we like to introduce some learning objectives because it's important to be able to order your learning. And some of the things we hope to achieve by the end of this session is that we should be able to define what good health is and know what can cause ill health. We should be able to describe the difference between communicable diseases and non-communicable diseases, and actually be able to give some very common examples of each. You should be able to understand that the presence of one disease can actually lead to a higher susceptibility of other diseases. And what we mean by that is one disease can actually make more likely for you to catch other diseases as well. We want to be able to define the term pathogen and know that it includes things like viruses, bacteria, fungi, and protists. We want to know how pathogens cause disease, and obviously different pathogens will cause disease in different ways. We need to understand the basic differences between a virus and bacteria, and know how pathogens can spread and human interventions that stop them from spreading as well. And obviously, we're going to look at some very common infections, including cholera, tuberculosis, malaria, HIV, and Ebola. My presentation seems to have frozen. Just bear with me 1 second. Right, let's start that again. Okay, so remember, we obviously tried to cover a couple of exam boards for you guys. So again, in your own revision time, have a look at what the specific exam board requirements are for yourself and order your revision around that. So the first question I'd like to ask you guys is, we're going to start off with a bit of a slider interaction. So for those of you who aren't familiar with this or are familiar with this, all you need to do is you need to join this poll at slido.com. You need to enter this little number here. It's quite self-explanatory, and then just pop your answers in. So what we are actually going to do during the course of this teaching session is I'm going to have a couple of slider interactions just to see what your guys' thoughts are on certain on certain topics. Now, the first question here is, what is a communicable disease, and can you give me any examples of it? And I am obviously going to check this with you guys, so it should work with no issues. I have checked this before. So again, all you have to do is you just go to slido.com and enter the code. Okay, so what is a communicable disease? So people are sort of coming in with their responses now. A disease that can spread from one person to another. Someone's given us an example, such as malaria. Someone said salmonella. Good. Any examples, anything you guys can think of, is to drop it in the chat. Diseases that can spread, a transmittable disease. Good. So remember, one of our learning objectives is to be able to define what communicable diseases are. Very relevant one. Someone has said a disease that can be such as COVID-19. Absolutely. A disease that is able to spread through different means, air, droplets. Good. A disease that can spread between two organisms. Someone has mentioned HIV. Excellent. Someone has said measles. Good. Yep, that's sexually transmitted infections. Someone has mentioned HIV and HPV, tobacco mosaic virus. Someone has also actually gone to explain that it's a disease that is spread through contact, not genetics. Excellent. Okay, fabulous. So a lot of you guys are saying the correct things, which is good. So the key thing here is, can you define what a communicable disease is? And what are some of the examples? It's very important to be able to say, this is a communicable disease, this is why it's called this disease, and be able to give some examples. This can be a very easy 1-mark question in your exam questions. Okay, next question. What is a non-communicable disease, and can you give any examples? So remember, here is where we're trying to differentiate between a non-communicable one and a communicable one. So someone has said a disease that cannot spread. Good. One that cannot be passed from one person to another. Good. So someone's identified cancer as being one. Excellent. A disease that cannot spread. Good. So the whole point and the key difference between these two types is the one is spreadable and the other cannot be transmitted. Good. So someone has said coronary heart disease, AIDS. Good. Genetics. So someone has mentioned something like Huntington's. This disease can't spread. So a very good definition has come in where someone has said it's a non-infectious disease that cannot be transmissible from one person to another. Some of these answers are very, very good and detailed. It's really good. And it definitely shows you guys have a very good baseline about what these terms are and what their definitions are. So we're going to move on. Good. So anybody saying heart disease, any genetic conditions? I think Huntington's and cystic fibrosis has come up. All of these are excellent. And as long as you're able to say very confidently that they cannot be transmitted from one person to another, you're on the right lines. So next question. What is the definition of a pathogen? And can you give me any examples? Again, we're just exploring baselines. So someone has said sickle cell. That's not very right. A disease causing microorganism. Someone's given an example, a harmful microbe, a microorganism that causes harm in the body. An organism that harms the body, bacterial pathogens. Can you give any examples of any bacterial pathogens you know? A bacteria that infects organisms, microorganisms that cause disease. So a lot of the same term is coming up, which is excellent, which means you guys know the standard definition. So myasthenia gravis is a genetic condition. It's not a path caused by a pathogen. Fungi, viruses, lactobacillus. Good, an organism that causes disease, viruses, bacteria, fungi. Bacterial infections. an organism that enters the host organism and causes disease. Fabulous. Okay, this is really good. So again, very simple definitions, because it's these definitions that are going to create the foundations of everything else we learn. Someone has talked about salmonella bacteria causing salmonella infection. Okay, fabulous. So thank you for dropping in your answers. That's actually one of the ways in which we're going to continue to interact with you guys during the course of the session, just to sort of break up the teaching a bit and to give you guys a minute to sort of put your thoughts out there about some of the topics we're discussing. So, what is health versus disease? Now, health includes mental, physical, and social well-being. When we look at a patient in medicine, we don't just look at their physical problem. We want to ask them about their mental health. We want to ask them about their social life as well, because these all play a very, very important role in making sure the person is okay and how functional they are. A disease is being unhealthy due to the malfunction of the body or mind, again, factoring in the fact that we can have physical ill health and we can have mental ill health as well. Having a disease is not the only form of being unhealthy. Poor diet, unfitness, and loneliness can make you unhealthy, but they're not diseases. So one thing we have to be able to establish is you can be healthy and you can be unhealthy. Now, you don't necessarily have to contract a disease from a pathogen to make you feel poorly. Factors like your poor diet, being very unfit, being lonely, again, talking about mental health, makes you unhealthy, but they're not diseases whatsoever. So, there is a wide range of different diseases, but we can often separate them into two groups, which is what we've just talked about. We can talk about infectious, communicable diseases, and non-infectious, which are non-communicable diseases. And as you guys have rightly identified, one, which is the infectious one, can be transmitted from person to person, and the other one is non-infectious and it's not transmitted from person to person. So, having a look at examples of infectious diseases. As you guys said, it's caused by other living organisms called pathogens. And then you very rightly defined pathogens for me and said a pathogen is an organism that causes disease. There are different types of pathogens, and the four main families of pathogens you guys are expected to remember are things like bacteria, fungi, virus, and we're going to look at protists as well. Now, when we talk about bacteria, we can look at an infection like cholera. I will tell you a little bit more about that in a little while. Cholera was an infection that happened very, very long time ago. It's not something where we have to deal with in this country now, but it caused a lot of death. Fungi, athlete's foot, COVID-19, we're all aware of. Protistas, which are otherwise known as protists, cause things like malaria. And parasitic animals such as tapeworm. So again, you guys may be familiar with the way I teach. I like giving you guys visuals. I like putting in stories to really help you remember some of the things I talk about. So here is a very, very wonderful example of athlete's foot. It's basically, reason it's called athlete's foot is because it's something quite commonly seen in athletes who wear trainers. They sweat a lot and it can make them more susceptible to getting fungus on their feet. It's very treatable with some antifungal cream and your foot is good as new, but this is what we refer to when we're talking about athlete's foot. It's a fungus of the toes. This little creature here is actually a parasitic tapeworm, and this over here is an example of a tapeworm being sort of unleashed from someone's body and sort of being excreted into the toilet bowl. Again, an infection of the tummy. It can happen. I mean, I don't know exactly what foods can cause this, but some infected foods, what you'd assume is any food that's infected with either the eggs of the tapeworm or the tapeworm itself, you consume it, you're going to have one of these beautiful things growing inside you, and it can make you very, very poorly. Okay. Now, a little bit about cholera. Again, it's going to help you guys remember. So remember, cholera is an example of a bacteria that causes infection, and it's actually quite a dangerous bacterial infection to have. It's an infection of your intestines, and what it does is it causes very, very severe disease and dehydration, and if it's not picked up and treated promptly, you could actually end up dying. Now, the way it spreads, and this is why it's so important, is people actually ingest food or water that's contaminated with the cholera bacteria, and often it's it can lead to very, very awful. So basically what the cholera bacteria does is, and a lot of bacteria do this, they release toxins into the patient's body, and the toxins actually stop the patient from absorbing water, digested food, and it basically results in the patient having chronic, like acute, literally really, really awful diarrhea, and lots of dehydration results, and the patient can become quite poorly. Considering we're actually living through a pandemic right now, it's only fitting to talk about previous ones as well. Now, we actually had a massive cholera outbreak in, I think, the 1800s, if I can read that correctly. And this is a map of England where we had all these cholera outbreaks. I mean, the cholera outbreak in London was actually quite awful. And back in the day, we had some very artistic newspaper letters going around when they started understanding what was going on. So remember, cholera is a bacterial infection that's caused often by contaminated water. And these are some of the posters that came out back in the day. So we had like the angel of death here, who was sort of pumping out water to the people because it was basically the death by water. And, you know, you talked about things like this, the silent highwayman. Again, it took them a while to realize how contaminated the River Thames was in London, and they often referred to it as a monster soup, and it was a cholera bacteria within the water that everyone was using, and that's what was making people so sick. So remember, back in the day, you were sort of excreting your feces and or your toilet into the Thames as well. Like it was basically monster soup, the way they very, very correctly described it. Okay. So again, a nice way to sort of remember what cholera does, the fact that it's a bacteria and that it's transmitted through water-based infection and food. Now, when we talk about non-infectious diseases, we're sort of talking about the other end of the spectrum. And remember, we've said that non-infectious diseases are caused by genetics, lifestyle, and other environmental factors. And the key thing is they can't spread. If you have cancer, you can't actually cough and spread it to somebody else. If you have a genetic condition, the only way it's going to be passed down is through your genetics. So common genetic diseases are things like cystic fibrosis. We can talk about environmental cancers such as lung cancer. It's a very, very common one associated with smoking. And we can talk about mental health diseases as well. Again, all of these things are not infectious in the sense that if one person has it, the other person will get it when they are in contact with person B. However, these are things that are impacted and caused by genetic factors, lifestyle, and environmental factors too. So just a little bit about cystic fibrosis. Yes, I'm actually sure you guys are well aware of this disease. It's the disease of the lungs, and it's actually caused by a genetic, if I remember correctly, I think there is a genetic mutation in the CTFR gene, which is actually a chloride channel in your lung tissue, but I'm not going to go into too much detail. That's just the medical nerd side of me coming out. But the thing is, the whole point of this is a genetic condition caused by a mutation in a very, very particular gene. And what ends up happening is, normally in a healthy individual, when you have your airways, they're actually very nice and open, and you're able to get air in and out of your lungs very, very easily. And there is no issue of removing debris or mucus. However, because of this faulty gene in our cystic fibrosis patient, it leads to increased mucus secretion, and you can't actually remove the mucus, and you end up with very, very narrowed airways, and the patient's lungs eventually turn into mash. It's actually quite an awful disease to have. And obviously, because of modern medicine, you're able to deal with it and you're able to live with it. But again, it is something that is caused by genetics, and it is actually, for anybody wondering, it's an autosomal recessive condition. So the thing we have to remember about diseases is having one disease can make a person more susceptible to other diseases. For example, when a patient has HIV, they're more susceptible to going into the AIDS state. And when a person is so immunodeficient, they can end up being susceptible to more bacterial infections. In fact, patients with HIV and AIDS don't actually end up dying from HIV itself, but they end up dying because the immune system is so compromised, they end up dying from other very basic infections. So remember, with HIV, the disease prevents the body's immune system from working. And we'll sort of look at HIV in a bit more detail later on about why it actually renders the immune system to stop working in the first place. We need to remember that some treatments actually increase the risk of other diseases. When we go through radiotherapy, which is actually the treatment of cancer using radiation, we can actually cause more cancers. And again, depending on the type of cancer it is and what cells we're removing, this can put the patient at more risk of other conditions. So remember, immune reactions to pathogens can cause certain allergies to take place. And viruses can cause cancers. And this was actually mentioned in one of the polls we did earlier on. Someone mentioned the HPV virus. Now, this is a human papilloma virus, and it actually causes cervical cancer. And this is actually very important because we actually now get a HPV vaccination. And just again, a little bit of context. The thing with cervical cancer is that if it's left untreated, it can turn into a very, very awful cancer that sometimes is too far gone to treat. And what they've now tried to do, it's all about disease prevention in modern medicine. Now, what we basically try to do is, we've obviously had the HPV vaccine that teenagers get in high school. And the whole idea is that it protects you against getting infections from the HPV virus. And if you've got pre-immune protection from it, you're less likely to contract the cancer. In fact, because of the way this cancer works, we now have cervical screening tests as well that can detect HPV in females, and this screening sort of happens past the age of 25, and it's a way of sort of examining the patient again and again and again over the years to sort of check for any changes that could potentially increase their risk of getting cervical cancer. And one of the key things is that it's be able to detect whether this patient has any HPV infection. The whole purpose of breast cancer screening as well, again, two different types of cancer. So remember, breast cancer isn't caused by a particular virus or pathogen. It's caused by genetics and environmental factors. But again, we have generalized screening for it as well to detect changes earlier on. So, the next question I have is another poll for you guys. How are pathogens transmitted? You guys told me we've got communicable diseases and non-communicable diseases, and we've already said that in communicable diseases, there's transmission occurring. But what I want to know is, how is this transmission occurring? What are some of the examples you know about pathogens being transmitted from A to B? So just drop your thoughts in this poll. So a very comprehensive answer coming through. Pathogens can be transmitted a few ways depending on the type. Skin contact, body fluids. So someone has said vectors, and you've given an example of malaria. Water and air particles, skin contact, excellent. Airborne. I mean, we're dealing with pathogen transmission at the moment with the whole COVID situation. So again, all this topic is very relevant to what's happening now in our day and time. So through water, soil, and skin-to-skin contact, pathogens can enter through the mouth, coughing, sneezing, air, sexual contact, contaminated surfaces, close contact, bodily fluids, air, water, direct contact, vectors. Vectors has been mentioned a couple of times, animal vectors. So remember, malaria is a type of condition or infection that happens through the vectors of mosquitoes. Coughing, contact with feces. So remember, not generalized contact with feces, contaminated feces is what we'd like to go for. Indirect transmission and direct transmission, good. That's a very good phrase to be using. Water and air contact, body, okay, good. So a lot of the generalized answers coming through, you guys are all sort of saying the same things, good. So remember, we're trying to build a profile here. What are pathogens? What are communicable and non-communicable diseases? Can you give examples of pathogens? And if we're saying pathogens cause infectious diseases, how do these infections spread? Good. Thank you for your answers, guys. These are all correct. I don't think I've seen a single wrong answer on there, which is good. Okay, transmission of pathogens. Different pathogens have different methods of transmission. There are two main categories of transmission. We've got direct contact. So this is physical contact between an infected person and an uninfected person. Again, something you guys identified. And indirect contact, where we've got no direct human-to-human contact happening, but there's like a third party involved, such as a dirty surface or a vector, etc. So let's have a look at some individual examples. Now, when we talk about direct contact, often what we've got is things like droplet transmission. Pathogen is spread through large droplets. So things like tuberculosis, if it's infectious in the patient's lungs, we often tell them to isolate because it's the type of thing that can be transmitted through large droplets. So remember, droplet transmission can occur through coughing, sneezing, talking, spitting, laughing, etc. Airborne transmission is basically when the pathogen is spread through via small particles in the air like dust. So these are things like spores. Now, spores is basically a type of way in which bacteria can sometimes sit and they can attach to dust particles and they can move through the air, which is why it's very, very important. And especially COVID has really brought this through about how important it is to make sure we're cleaning our surfaces, our worktops, anywhere we go, we're cleaning surfaces all the time to reduce this airborne transmission. Transmission of pathogens through vectors. Now, this is an example of where a pathogen is spread through another organism, normally animals. And I've given you guys a very, very vivid picture of a mosquito here, just to emphasize that mosquitoes don't cause malaria. They are the vector carrying the malarial pathogen. So mosquitoes and rats are all examples of pathogens that are transmitted via vectors. And it's quite important to be able to identify what sort of disease is actually spread through each one. So mosquitoes, again, the first thing that comes to mind is malaria. Ticks, I'm going to tell you a little bit more about. One of the common diseases that ticks cause is Lyme disease, and rats as well are to do with the plague. Okay, so again, I'm going to give you guys a bit of more fun facts about history of medicine in a second according to these vectors, because I think some of these sort of anecdotal things will help you remember that it's not the actual animal itself carrying the pathogen. So often with malaria, mosquitoes, what's happening is the mosquito is infected with the malarial protoctista. And what then happens is it's going to go and bite the human. It's going to sort of nibble on its skin and, infect its blood. And then that pathogen is then in the human's circulatory system. And they've made the patient very, very sick. So in regards to ticks, now over here is a tick. It's literally the tiniest thing ever, but can cause the worst thing ever. Now, again, one of the things, so the tick, again, isn't actually causing the disease. It's the tick carrying a pathogen, and it can actually give you something called Lyme's disease. And it takes a while to sort of develop once you've actually been bitten. And the patient will present with things like fevers, chills, headaches, they'll be very tired. And then eventually they'll have some more sinister signs such as dizziness, irregular heartbeat, and they'll have some facial sort of distortion as well because of damage to nerves, etc. Now, one of the ways in which clinicians are able to identify Lyme's disease is by this very, very prominent rash that occurs at the site of infection called a bullseye rash. And you can see why it's called a bullseye rash because of the way it looks. And the minute we see this and we've suspected they've been somewhere where they're very susceptible to things like tick bites, we want to give them some antibiotics straight away because this can make them very, very poorly. Now, again, another example of where animals, the vectors, we all, I'm sure, at some point in our journey through high school, we've learnt about the plague. Now, historically, it was believed that the disease was an infection of the bubonic plague was basically being caused by rats, but it actually took them a while to realize that it was fleas sitting on the rats or biting the rats that was then being spread to humans. So basically this picture, I wish this was a bit bigger, but you can see basically the fleas is sitting on the rat and it's the fleas that are carrying the microorganism, the pathogen that's infected the rat. And we knew rats were basically running loose on London streets back in the 1800s, etc. And they were then transmitting it and spreading it. And it was actually a very, very awful epidemic, and it spread like wildfire in the 1600s in England, and many, many people died because of it. Okay, just again, emphasis on the idea that pathogens don't have to transmit infection directly. A lot of these are happening through vectors. So then the other example we can talk about is direct contact, and this is when we have contact with an infected person. I think a common one is basically things like the flu, when and it's all about when you have the flu, wash your hands again and again, because if you've got germs on your hands and you go and shake hands with another person, you're going to transmit the infection. Hands, so you know, rashes can also are a good way to spread disease from an infected person to a non-infected person. Now then we talk about common vehicle transmission, and this is something again you guys mentioned in the poll. So this is basically where the pathogen is spread through contact with an infected object. So things like clothes, tools, food, water, etc. Okay, and it's the idea that other people are going to go and then use these sort of clothes and food and water, and then we're going to end up spreading the infection. And cholera is a very good example of this. So one of the other things we forget about is, and this was actually more relevant back in the day when living conditions were really awful, is that conditions do affect transition rates and transmission rates. Now, one thing we have to remember is this is, again, something we talk about during the times of COVID. One of the reasons we've implemented social distancing and we stopped large gatherings is because tight spaces, lots of people was going to increase the risk of transmission. So high population density is a type of living conditions that was going to really infect people quite rapidly. Dirty, unhealthy, unhygienic conditions, humid conditions as well. Remember, it's warm, perfect environment for pathogens to grow. And high vector populations as well. So again, basically things where we've got areas where there's a lot of rats and lots of mosquitoes, etc. You're asking for disease transmission. Okay. So again, another poll coming up, introducing the next sort of thing we want to talk about. A very, very simple question for many, but what I would like to know is, are viruses living things? True or false? We're keeping it nice and simple. And I personally am expecting 100% on this. So are viruses living things? And although I'm not asking you guys, this is an open-ended question, I want you to think about why you are putting the answer down you are putting down. What are your reasons behind it? Because often in your exam questions, they're not going to sort of just say to you, they're not going to just give you a statement and say true or false. They're going to give you a statement and say, can you give reasons why? So I'm just going to wait for some of you guys' responses to come in before I sort of release the outcome of the question. So again, I'm personally expecting 100% on this answer because I think this is something we talk about quite regularly. Just give everybody a chance to sort of put in their responses. Okay, we've got a solid amount of responses coming in. So are viruses living things or not? So remember, okay, so we've got a bit of a 50-50 here, which was slightly unexpected. So viruses are living things. Viruses are not living things, okay? I can't believe there's a 50-50 divide on this, but that's fine. That's the whole purpose of these revision sessions. So one of the reasons why viruses aren't classified as living is because they cannot actually survive without a host. So remember when we talk about viruses, in order for a virus to duplicate and really flourish in its it needs to basically have a host. Okay, it needs to go and infect something before it to be living again. A virus can't go from 1 to 2 without actually replicating in another cell. So this debate about whether viruses are living things is something that comes up, and it's important to establish the reasons why. Now, another question, we're looking at bacterial cells, and as you can see over here, we're sort of trying to compare common pathogens from one another. Do bacterial cells contain no DNA? True or false? Sorry, let me say that again. Bacterial cells contain no DNA. Is this true or is this false? Just give you guys a minute to give all your options. Okay, just a couple of more seconds more. Okay, good. So this statement is false. Bacterial cells do contain DNA. Remember, it's going to be encapsulated in a capsid in the middle of the bacterial cell. So again, this is introducing the next thing we're going to be talking about, is where we look at viruses, where we look at bacteria, and we look at the differences and we look at different examples of these. So, can you name any bacterial infections? And I'm expecting lots of responses about really, really cool bacterial infections here. So, cholera, good. What else do we have here? So, guys, remember COVID-19 is a viral infection, not a bacterial infection. We've got salmonella coming in. Let's see what else comes through. Cholera. So apart from cholera, does anybody know any infections? Oh, okay, we have a list. Strep, UTIs. Okay, so that's obviously gone down, but there was a very big list coming through there. Tuberculosis, excellent. Gonorrhea, good. Meningitis. So meningitis is actually one that can be caused by viruses and bacteria. Rose black spots. Food poisoning can be caused by bacteria. Yep. Pneumonias can be caused by bacteria. Excellent. Okay, good. Fab. So again, lots of common bacterial infections out there. Okay, so MRSA, tuberculosis, good. So a lot of the common bacterial infections coming up. Fabulous. Now, on the other hand, again, it's all about distinguishing between viruses and bacteria here. Can you name any viral infections? COVID-19, apart from COVID-19. Tobacco mosaic virus, thank you. HIV. The flu. Malaria is actually caused by a protist. Someone saying dengue. Tobacco mosaic virus, HIV, COVID-19, Ebola, measles, rubella, chickenpox, smallpox. Impressive. Okay, good. Fine. HPV, good. Yep, measles. Okay, excellent. So good examples of viruses coming in. Someone has said the common cold. So when we talk about viral infections, I am actually going to go through the differences between HIV and, sorry, not HIV, the common cold and the flu, because it's important to be able to distinguish between the two. Okay, yep, virus, exactly. Good. So again, very, very correct responses coming through. Thank you. So thank you for your responses. What we're going to do now is we're going to actually look at bacteria, and we're going to look at viruses, and we're going to look at some of the common infections these cause. Now, as you can see over here, this is a picture of a bacteria. Remember, bacteria will contain DNA. In this scenario, in this particular diagram, they're encapsulated in a nucleus. So remember, bacteria have plasmid through which they can transmit their DNA. They have ribosomes, they have capsules, they have cell walls, they have a plasma membrane, and they have flagellum that helps them move. Now, bacteria, one of the reasons they're living things and classified as living things is because they can reproduce rapidly. They can release toxins and make you very, very poorly, and they damage tissues. And they're quite small, they're between 0.5 to 5 micrometers in size, but they can cause a lot of damage. Now, one of the infections we discuss here is tuberculosis. Now, tuberculosis is an infection of the lungs caused by the Mycobacterium tuberculosis bacteria, or Mycobacterium bovis bacterium. Now, the transmission is actually via droplets. So this is why a patient who has tuberculosis, and it's the infectious type, when they cough, they're going to get TB everywhere. So this is that often when we have patients in hospitals with TB, we sort of isolate them in an individual room. So some of the symptoms of TB, you can have a persistent cough, you're going to have weight loss, you're going to have night sweats, you feel very tired, you may have some neck swellings because of the lymph nodes in your neck swelling up, and you have a loss of appetite. So you become very, very poorly. And one of the ways in which we can detect it is by doing lung biopsies and lymph node biopsies to identify whether this particular bacterium is present or not. So again, the thing with TB, and TB again, not such a big problem in this country, but can be a big problem in sort of third world countries, is that oftentimes there are 3 different types of TB. You either have a patient who has no TB, or you can have a patient with active disease, or you can have a patient with latent TB. Okay, now one of the problems with tackling TB is that patients who are affected may have no disease symptoms. And basically what happens is, and we see this a lot, some patients, so say for example, someone is born in a country where TB was quite prevalent back in the day when they were born, they can actually carry around the TB bacterium in their lung tissues for a very, very long time. And the only way it becomes present and active is, say, for example, if the patient was poorly or they became immunocompromised and their immune system wasn't as robust as before, then it can turn into an active disease. And this sometimes makes it very hard to find and identify people to treat. So Salmonella is another infection we talk about caused by the Salmonella bacterium. Again, the common vehicle is food. Symptoms: stomach pain, vomiting, profuse diarrhea, and you become very, very sick. So some of the common things that this bacteria can be found in is things like pork, chicken, eggs, cow milk, etc. The type of bacteria is actually a gram-negative bacteria, and it's basically transmitted during contact with infected pigs, cattle, and pets. Okay, so these are the sort of things that will cause salmonella. And it's not like you basically go and eat the cow itself, it's the milk that's going to contain it, or it's the meat of it's the pork meat, or it's the eggs that are going to contain the infection. Viral infections. So remember, the key point here is that virals are not classified as living things. While they have some genes and they have all these fancy things in their actual molecule itself, the thing is a virus cannot survive on its own. It has to go and infect another cell. It has to take over the cell's machinery to then survive and reproduce. And one of the things that we need to be classified as living things is the ability to reproduce. So viral infections use host cells to replicate. They hijack the replication machinery, such as your ribosomes, and basically once it's an active viral infection and they have sort of hijacked the replication machinery, they replicate themselves and basically make baby viruses, and enough baby viruses to cause the host cell to burst and become damaged. Now, you can have active viral infections, or we have latent viral infections that lie dormant, which means it just sits there in the background chilling and not doing anything. A very good example of these two types of one infection is basically chickenpox. An active chickenpox disease occurs when you are a child, so when you're in like you're 8 to like 10 years old, or if not smaller. And obviously it's caused by the varicella zoster virus. Now, chickenpox in adulthood or late adulthood, when you're sort of in your late 50s, 60s, it depends, can actually come back as something called shingles. And that's where and it's a very good example of the virus being latent. So what basically happens is you have the virus, and some of the viruses actually stays in the body and says, you know what, I'm just going to hang back. I'm not going to do anything. I'm just going to sit here and chill. And for some reason, you can have the reactivation of that particular virus, and it comes back as shingles. So again, the whole question is here, they're tiny, they're simple, but are they alive? They're not alive. They're not classified as technically living things. HIV and AIDS. So remember I talked about, and you guys did identify HIV. So remember we said that HIV is known as the human immunodeficiency virus, and its transmission is through bodily fluids, and we can actually detect the viral load in the patient's body as well. Now, the thing with HIV, and as what all viruses do, it actually uses white blood cells as a host. So it doesn't just go in and walk around the patient's blood, it will infect the white blood cells. And we all know white blood cells, and we will all learn today later on in the day that they are very, very essential in the immune response to any infection. So what basically happens is you have infected white blood cells that your immune system gets rid of because they're infected. Then we end up with no white blood cells, and we end up in an immunodeficient state. And like I mentioned earlier on in this session, is that people with HIV actually die of other diseases as opposed to HIV itself because of the damaged immune system. So again, a very, very important distinction to make. So as I was saying, HIV is the virus which attacks the immune system in humans, and AIDS is a medical condition when the immune system becomes too weak to fight off infections. So one of the ways in which HIV can be transmitted is through the use of non-sterile syringes, pregnancy and breastfeeding, blood transfusion, organ transplant, and unprotected sex. And unprotected sexual intercourse is the biggest and most commonest way in which HIV is passed on. So common facts about HIV that people often aren't aware of, as we have said, people on effective HIV treatment can't actually pass it on because the viral load will be very, very low. Okay, it can't be passed on through day-to-day contact. Any of these examples are not examples of day-to-day contact. By day-to-day contact, we mean handshaking and talking and coughing, etc. Okay, and it can affect anyone. Okay, so then there are some people that are more susceptible to getting it, but it can basically affect anyone. And there are many ways in which to prevent it as well. So, getting regular tests, never sharing needles, using protection. And obviously, if you are living with HIV, making sure you're taking all your medications on time. So, viral infections. Now, when we talk about viral infections, we can talk about the flu. Now, when we talk about the flu, there's actually two different types of flu that we should be able to distinguish between. In the winter, when you have the sniffles, you have something called the cold, okay? And that's caused by the virus called rhinovirus. When you have a type of cold where you are so miserable, you have a temperature going through the roof, you feel shivery and you can't move for a couple of days, you were infected with the influenza virus, okay? So the flu itself is caused by the influenza virus, and this is what we are vaccinated for against every year. The virus actually evolves very, very quickly, and it changes its antigens and glycoproteins all the time, which is why we actually need a new vaccine for it every year because of this quickly changing. So basically, a flu virus that was there in 2017 will be different to the one in 2018, and it will be different to the one in 2019. And because it's different, it means you can get infected again. And the flu itself does actually kill. So remember, the cold or the flu. So the common cold, which is caused by the rhinovirus, is the symptom onset is very, very gradual. You never get a fever. You're a bit achy, but you don't get things like a headache, and you don't feel tired and fatigued. But when you get the flu caused by the influenza virus, which we're being vaccinated for, you become very, very unwell very, very quickly, and you can end up in hospital with this. So again, the influenza vaccine we get every year. Ebola, again, another infection that was basically a bit of an epidemic a couple of years ago. It's caused by the Ebola virus, and this is what the virus looks like. It's got a very, very high death rate because it basically causes a hemorrhagic fever where you basically end up bleeding and you're feverish, and it's quite awful, transmitted through body fluids. Fevers, body aches, you've got internal, external bleeding. So we call it hemorrhagic, basically means to bleed. You've got a fever and you're bleeding from everywhere. So you become very, very sick very, very quickly. And again, I was referring to the Ebola epidemic. Now, Ebola actually, when we were talking about Ebola in the news, it was around 2014, and it was the biggest outbreak in West Africa. Now, again, something to bear in mind is that Ebola, basically, the first outbreak of Ebola actually happened back in the 1900s in 1976, okay? And the virus has obviously been present in the world for some time, but we had the biggest outbreak in Africa in 2014. It was the same with like COVID, like we had the SARS virus and a couple of other viruses before COVID-19 happened, and it kind of just wreaked havoc everywhere. So again, this is what I was talking about in terms of common symptoms: fever, weakness, muscle pain, headaches, and a sore throat. It spreads through contact with bodily fluids, including dead bodies. So bodily fluids, we mean things like blood, feces, vomiting. It's not airborne. So remember, influenza and tuberculosis was airborne. COVID-19 airborne. We're putting face masks on. With Ebola, it wasn't spreading through the air. It was spreading through actual infected body fluids. Okay, and although it's a fatal illness, if the patient is given good medical care earlier on, they can reduce the risk of death. Protoctista infections. Again, this is what Protoctista looks like. You don't need to worry too much about the detail about what's actually present. Just know one of the most common things is that it enters and feeds on cells. Now, the organisms can be a mix of bacteria, plants, and animals. And basically, pathogenic Protoctistas that are actually infectious can be very, very small. And a common example is that of malaria. So malaria, remember, again, I showed you that mosquito with the pathogens inside the tummy of the mosquito. The mosquito bites the human. I'm losing my voice. The mosquito bites the human, and then the pathogen, the Protoctista, is going to be unleashed into the human's blood. It's caused by Plasmodium falciparum, so that's the actual pathogen, and it's transmitted by vectors, such as the Anopheles mosquitoes. Now, in terms of the life cycle and how it spreads, is the mosquito bites an infected person who already has it. So basically what happens is the Plasmodium gamete, the pathogen, enters the mosquito. The gametes fuse and develop into zygotes, and they basically travel into the salivary glands of the mosquito. This infected mosquito goes and bites an uninfected person, and now the uninfected person has become an infected person. So again, just emphasizing the idea that it's the mosquito carrying the pathogen, and it's being transmitted through vectors. Okay. Fungal infections. Again, I showed you athlete's foot. Here is an example of a fungal infection called a ringworm, and it's actually caused by 40 different types of funguses. It can be transmitted from touch, so if someone has it and they touch you, you can get the fungus, or it can be done through dirty clothes, etc. Common infection of human skin and nails. You get this red, itchy, circular rash, and the rash is very, very contagious. Again, very treatable with some antifungal cream. So I've now got a little bit of a quiz for you, just to sort of bring in some of the concepts we have talked about. So again, we're going to go on to the go on to Slido, put your name in, and sort of join the quiz. It's kind of nice to sort of see, I think we have like a leaderboard at the end, and it can sort of show who's doing well and who's answered the most questions right, etc. So question number 1. Which pathogen is an example of a virus? Again, very easy question. These are sort of things we have covered already. So the first question is, which pathogen is an example of a virus? Is it Salmonella, Ebola, ringworm, and malaria? And again, when you're looking at these, look at the questions and be able to identify each of these pathogens. You should be able to say from the top of your head which one is which. So in this example, remember Salmonella is going to be, think about what kind of infection it causes. Think about what we talked about Ebola. So one of the reasons I show you pictures and I tell you mini stories is because it will help you remember the infection. I want you to be able to look at these very confidently and be able to say, right, this is caused by this. So again, a lot of you guys have said Ebola. So remember, Ebola is the one that's the virus that I was literally just talking about 2-3 minutes ago. Salmonella is caused by a bacteria. It's a bacterial infection. Ringworms are caused by funguses, and malaria is caused by a Protist. Remember, we were called to a malaria falciparum. Okay, so remember, be able to distinguish between these. So for those of you who said Ebola, very well done. So we can obviously see someone answered this in less than 10 seconds and got it right, so well done. So really do participate in these. It's nice to sort of see how you guys are doing. So again, which pathogen is an example of a bacteria? Again, I'll give you guys a couple of seconds to do the answer. It's important to be able to look at these and be like, right, I know what the infection is. I know a little bit of background about how the infection comes to be. So really use these questions as a way to test yourself. I actually think you guys, I mean, I've got the thing open on my phone and I actually don't think okay, this is a mistake of mine. I think the voting wasn't open for this one. Actually, you guys have been able to do it. Okay, so for any of you guys who selected Salmonella, good. I'm not too sure that one was actually, you were able to vote on that one, but you should be able to do it on this one. So which type of pathogen requires a host cell to survive? Bacteria, viruses, or yeah, okay, your things are coming through. Okay, I apologize for the glitch on the previous question. Okay, so remember, one of the things we said that viruses are basically non-living organisms and they require a host cell to survive. Remember, we had said that they were going to infect the host cell and basically cause havoc by taking over the replication machinery and then make baby viruses, cause the host cell to sort of burst open and the virus is released into the host body. Fabulous. So environmental and genetic factors cause which type of disease or contribute to which type of disease? Again, looking for 100% on this one because it's very, we talked about this quite a lot and I think it's quite straightforward. Just wait for your responses to come through. Okay, so remember the correct answer is non-communicable diseases. So remember we said that non-communicable diseases were not infectious, things like cystic fibrosis, we talked about breast cancers. Communicable diseases are the one that are infectious and you can spread the infection from one to the next. So for all of you who said non-communicable, well done. Okay, which pathogen causes the common cold? Again, kind of expecting 100% because I literally talked about this less than 5-10 minutes ago. Okay. So the correct answer for the okay. So this is interesting. Now, again, I'm going to make this distinction, and this is why some of these questions are very, very useful to clear up confusion. So remember, I said there were two types of colds. One was the common cold and one was the flu. Now, when we talk about the common cold, we're talking about the rhinovirus, which is going to give us the little sniffles and we're going to feel a bit down in the dumps. The flu is basically going to be caused by the influenza virus, and that's the dangerous one. That's the one that gives you the fevers, the shivering, it makes you really, really lethargic, and it makes you very, very poorly. Next question. HIV virus infects which part of the body? Is it the red blood cells? Is it the white blood cells? Is it the skin? Is it your liver? What do they infect? Again, it's important to understand how some of these viruses work and why the patient becomes so very ill. White blood cells. Fabulous. And remember, I mean, the specific type of white blood cell this actually affects is your CD4 T cells. And one of the ways, so remember we were talking about how because your white blood cells are infected, your immune system gets rid of the white blood cells and you end up with no white blood cells eventually, and you can enter the immunodeficient state. So to summarize, infectious diseases are diseases caused by other organisms, and they are known as pathogens. And again, you guys have been able to define that pathogens are things that cause disease. You can have one disease and it can lead to you having more diseases. And we've already said that non-infectious diseases are caused by environmental factors and genetics. There are different types of transmission: droplet, airborne, vector, direct contact, common vehicle. And some of the pathogens we've talked about are things like bacteria, viruses, protista, fungi, and parasitic animals. And these are all, and you should be able to identify individual examples of each. So remember, bacteria, salmonella, viruses, HIV, Ebola, flu, common cold, protista. We're talking about things like malaria. Fungi, think about athlete's foot, think about ringworm. Okay. Now we have a handful of exam questions now to do, but it's 11 o'clock and I think now would be a very good time to take a break. Okay, so I'm going to give you guys 5-minute break. It's actually 11:02, so what we will do is we will resume back at 11:07. Take 5 minutes. Again, we've covered a lot in this session already. I've thrown a lot of information at you, but you guys have done some really good interaction via the quizzes. So we're going to take 5 minutes, step away from your screen, go stretch your legs, go have a quick snack, and then we will come back in 5 minutes or so, okay? I'm aiming to resume by 11:07. So I'll see you guys in 5. Okay, I was on mute, never mind. Welcome back. I hope that was a refreshing 5 minutes for you. It's always really, really important to step away from your revision for 5 minutes or so, just to give your sort of brain a minute to sort of process everything it's just covered. So as always, we've covered a bunch of content, so we're going to explore some exam questions and just sort of get a flavour of what the examiners are expecting us to be able to answer in respect to this particular module. So we're going to crack on with some exam questions and then we will move on to the next part of the content. Just going to check my screen is all good. Okay, fab. So question number 1. It says scientists investigated the effect of different factors on health. People who are not active may have many health problems. The graph shows the percentage of 16-year-olds in some countries who are not active. So over here, again, always take a minute to look at the data. We've got on the y-axis percentage of 16-year-olds who are not active. And then we've got different countries at the bottom. Now it says what percentage of 16-year-olds are not active in the UK. So again, this just takes a little bit of table and graph reading. So what we can see over here, and I've already highlighted this, is here is the UK. So all we need to do is accurately read this bar graph. Now, what we can see over here is this is at just above 60. If we were to figure out the individual values of each of the intervals, we would realize that it's 2, so 62, 64, 66, etc. And looking at where the bar chart is, we should get a reading of 64. Now, what percentage of 16-year-olds in the UK are active? Well, again, this just sort of comes back down to a little bit of math. So remember, if the amount that are active is 64, then that are inactive is 64 and it's out of 100. All you're doing is you're doing 100 minus 64 to get the percentage that are active. Again, two very simple data interpretation marks. Now, part A, says a newspaper headline states that people in the UK are the laziest in the world, which is quite offensive. Information in figure 1 does not support the newspaper headline. Suggest one reason why the newspaper headline may be wrong. Other than the fact that it's completely wrong, look at the statement. It basically says people in the UK are the laziest in the world. First of all, the world has more countries than France, Germany, Italy, and Netherlands. Secondly, it says people. By people, we mean the whole population. But what is this particular investigation looking at? 16-year-olds. So therefore, you don't have enough data to support that statement. So again, making references to any of these types of points that only considers 16-year-olds. It does not refer to all ages. And there's only some countries, and it does not refer to all countries, the data, so you can't make these assumptions. Part B says doctors gave a percentage rating to the health of 16-year-olds. The table shows the amount of exercise 16-year-olds do and their health rating. So again, what we have over here is we have the amount of exercise being done in minutes and the health rating as a percentage. And what we can see is the more exercise you do, for example, at 300 minutes every week, the healthier you are. What conclusion can be made about the effect of exercise on an individual's health? So as you can see, it's only 1 mark, so they're actually just expecting you to make a very, very simple inference from the data. And we can see here, the more exercise you do, the higher the percentage of health rating, okay? And the more exercise is done, the healthier a person is. So it says inherited factors can actually also affect an individual's health. Give one problem that may be affected by the genes someone inherits. So being malnourished, not necessarily, and having a deficiency disease can actually be down to diet more than anything else. But having high cholesterol is actually something that's very much affected by your genes. And there is a condition called familial hypercholesterolemia. Again, you know, I'm big on putting things into context. It's called familial hypercholesterolemia, and it's basically a genetic disorder that causes very, very high levels of bad cholesterol in your body, and basically you can become very, very poorly with it. And one of the reasons it's so important is because if you have a family history, you're more likely to have early heart disease, and you could end up becoming very, very poorly. If you have heart disease, you're in big trouble. And obviously, to sort of prevent it, it's manageable. Obviously not preventable in the sense that if it's being passed down from your genetics, it's basically controlled by things like statins and different medications that collect and prevent buildup of cholesterol in the body. You can actually remove cholesterol from the blood as well through different techniques and lifestyle changes. So again, this is basically where they are sort of getting this type of question from. No, question number 2. Lungworm is an infection. Lungworm can kill dogs. It's caused by a small worm. The diagram below shows the life cycle of a lungworm. And what we can see here is that basically the lungworm reproduces in the dog and infects the lungs. The larvae, which is the young stages of the lungs of the worm, are coughed up from the lungs and swallowed. Fine. Now basically the worm larvae pass out in the feces, and obviously now the feces, the dog's poop, is on the floor. It's going to be picked up by snails. Then another dog is going to go and eat the snail, and then it's going to spread. Okay, so remember the whole thing here is, it's very interesting how it's spreading. A dog isn't eating another dog's feces. It's the idea that the dog is basically going to excrete the larvae into its feces. It's going to be picked up by snails, which are then carrying it, and then this is going to be picked up by another dog. Now, the type of organism this is representing is a vector. Okay, because it's being carried through 2-3 vehicles. Now it says suggest how the spread of lungworm can be prevented. So anything along the lines of what's spreading it. So basically any infected dog, you can isolate them. You can sort of bring up snail pesticides, so to remove the snails, okay? And make sure there's no infected snails there either. So any of these sort of condition points will work. So destroy the snails, isolate the infected dogs, treat infected dogs as well. And the key thing here is actually, and this is a very good one that I didn't mention, educate owners about picking up dog feces. Now, why is it actually, like, I think it's against the law to leave dog feces there. You can get fined if you don't pick up your dog's feces. So this is very, very important. You need to be educated about these things. Now, malaria is a disease spread by mosquitoes. Describe 2 ways to control the spread of malaria. Now, remember, malaria is spread by mosquitoes. Basically, you can have mosquitoes. I think if you go to like hot countries where mosquitoes are quite a problem, you can have mosquito nets and mosquito repellents that can help, and you can basically just make sure you're being very, very careful in terms of putting on a lot of anti-malarials on yourself as well to prevent the mosquito bite and prevent the infection spreading to you. So anything like stop mosquito breeding, so, you know, basically getting rid of mosquitoes and using mosquito nets and insect repellents, etc. It's going to get you the marks. Again, very, very simple concepts being tested. Again, a little bit about it. So malaria can actually make you quite unwell if you are to get it. And this is why when you go into countries or travel to countries where malaria is quite prominent, you're expected to take a course of anti-malarials beforehand. So it's actually estimated around 198 million cases occur each year of malaria, and loads of populations are at risk. And some of the common ways in which we can prevent it is by treating mosquito nets. So basically you have a mosquito net and it's sprayed with lots and lots of repellent. So not only do you catch the mosquito, you kill it too. Okay, and obviously as hard as it is, but in a hot country, you're not going to get bit if your arms and legs are coloured, are sort of covered, because then you're not going to be able to get it. Now again, the whole idea is that prevention is better than treatment. So basically actually keeping an eye on the weight in these preventative measures can basically really reduce the spread of it. Now, can't remember exactly what year this is from, but again, we may be just talking about a common condition, but loads and loads of people are killed by malaria. So 1 million are killed every year. 900,000 of these are young children. And there's actually only 2,000 reported cases of malaria each year, but it's the idea that it's imported. Okay, it's coming from somewhere else. So again, a common condition that can become very, very deathly if put into the wrong population. Now we're talking about Salmonella. So this is one of the reasons why I encourage you guys to be familiar with these different types of bacteria, because once you know a little bit of background, when you see them in your exam questions, you're not going to be taken back by, oh my goodness, what is it asking me to do? So question 3A says 2 symptoms of infection by Salmonella are vomiting and diarrhea. What causes these symptoms? Remember, we were saying a lot of bacterial infections actually cause the infection and make the patient poorly because of its toxins. Okay, it releases poisons and toxins that makes the patient very, very unwell. Give 2 ways in which a person with mild infection of Salmonella can help prevent the spread of the bacteria to other people. And I've given you a nice little infographic here. Make sure they're clean. They're washing their hands. We're cleaning surfaces. We're sanitizing. We're reading about how to prevent the spread of infection, and we're making sure everything around us is clean. So any sensible suggestion of how to reduce infection spread is going to get you the mark. Okay, so wash hands after using the toilet. Wash hands before preparing the food. If you are infected, don't prepare the food. Okay, and isolate yourself and disinfect clothes and surfaces, and don't share utensils and cutlery if you're infected. Okay, so any sensible example of keeping clean and hygienic should get you the mark. Now it says in a very serious infections of Salmonella, a doctor can prescribe antibiotics. Again, something we will talk about later on. It says which type of drug can the doctor prescribe to kill bacteria. So antibiotics, and I will probably say this a lot of times today, are used to kill and treat bacterial infections. Okay, just remember the B. We cannot use antibiotics to treat viral infections. Now it says a person with AIDS may take longer than a healthy person to recover from Salmonella. Explain why. So remember, if you remember what I said about HIV, so remember we said, say for example, these are your white blood cells, and this one is infected, this one is infected, this one is infected, and this one is infected. Now remember, it's infected with the HIV virus, so your body says, right, you're infected, I'm going to get rid of you. I'm going to get rid of you, I'm going to get rid of you, and I'm going to get rid of you. And once a patient has very, very few white blood cells left, remember, their HIV basically turns into AIDS, so immune deficient state. So now when we have a little virus or a bacteria sort of come in, they only have, in this very, very simplified scenario, 1 white blood cell to deal with the infection. Is that going to be enough for them to deal with the infection? No, it's going to take them much longer, and in fact, it can actually kill them as well. So the points you're trying to make here is the idea that the patient has a very, very weak immune system, and they don't have enough sort of firepower to deal with this infection. Okay, so the white blood cells cannot kill the bacteria because there's such few of them. Now it says Salmonella can be transmitted from chickens to humans. Chickens can be vaccinated to prevent the transmission of Salmonella bacteria to humans. Suggest one other way farmers could prevent the transmission of Salmonella from chickens to humans. Well, if you have 1 infected chicken, get rid of it or completely isolate it. Make sure the chickens are growing in a hygienic conditions. Okay, so anything like that, give chickens the antibiotics. Don't sell infected chickens. Keep chickens isolated, or slaughter the infected chickens. Okay, so things like that are going to be important to remember. Next part of the question says, a restaurant owner employed a scientist to test the effectiveness of 2 kitchen cleaning liquids. Now it says the scientist took samples from 2 work surfaces before the surfaces had been cleaned and after the surfaces had been cleaned, and the results are shown in figure 2. So again, number of bacteria down on the y-axis, and we've got the different types of cleaning liquids on the x-axis. So we can see over here, they've used 2 types of cleaning liquids, and the question says which cleaning liquid is more effective. Now, as you can see, it's cleaning liquid B that has the biggest decrease in the number of bacteria after cleaning, and we can see that from the difference in the height of the bar charts from before cleaning to after cleaning. So all you're saying in this answer, again, it's only 1 mark, so it means you're expected to make a very, very simple deduction. And it's the idea that cleaning liquid B results in greater reduction of bacteria in both locations. Now it says the scientists investigated the effect of cleaning liquid A and cleaning liquid B on salmonella grown in bacteria. So what we basically have here is a little petri dish, and what we can basically see is we've got a filter paper disc soaked in cleaning liquid A and a filter paper disc soaked in cleaning liquid B, and the petri dish itself has the agar, which is got the bacteria growing on it, and we've got a filter paper disc soaked in water as our control. Now it says the petri dish was placed in an incubator of 25 degrees Celsius for 48 hours. After 48 hours, the scientists calculated the area around the paper which had no bacteria growing. Now the idea is that the bigger the area, the better it is at destroying the bacteria. So we can see over here cleaning liquid A, cleaning liquid B, and water. And they're looking at the areas around the disc. It says, what measurement would the scientists need to calculate the area where no bacteria were growing? So basically, to calculate the area where no bacteria were growing, they would basically need to take the radius calculation, isn't it? So of the entire petri dish, and then minus it from the area with no bacterial growing. Okay, so it's the whole idea is that you've got the area, and to calculate the area, it's the whole idea of calculating the radius of it. That's it. Okay, and to put it simply, you're basically looking at the size of the area that's been cleaned. Now it says give 1 change to the investigation that would allow the scientists to check if the results are repeatable. So what ways in which they can do is basically repeat them and look to see if the results are similar. Okay, and it's a very, very simple question to ask. So remember, we always say in experimental results and experimentation, we say that in order for an experiment to be reliable and the results to be reliable, you should be able to repeat them and you will get the same results again and again and again. And that would basically, in this scenario, as we can see, cleaning liquid B had the biggest area around the disc with no bacteria growing in it, right? So what that would mean is if we were to repeat this experiment a couple of times over and we still found that it was cleaning liquid B producing the biggest area, we would say our results are repeatable and our investigation is sensibly put and constructed. Now it says the scientists showed the results to the restaurant owner. Both cleaning liquids cost the same per decimeter cubed. Suggest 1 other factor the restaurant owner should consider when choosing which cleaning liquid to use. Now, bear in mind that this is a restaurant and we're spraying chemicals onto surfaces. We want to know, is this chemical going to be dangerous to food? How does this chemical interact with other chemicals they may also be using in the restaurant? Is this going to damage any furniture? So, you know, any interactions this chemical is going to have, is it going to cause any damage to the environment it's being used in? So loads of different examples. So some of the things we can talk about is the toxicity, effects on other types of bacteria. Does it have any interaction with other cleaners? So again, you just 1 mark. Any sensible thing about interactions with other things is going to get you the mark. So that brings us on to the end of the first part of the lesson, and we've covered a lot and I feel like I've been talking loads and loads and loads. So just to review, we should now be able to we should now very comfortably, oh, hang on a second. We should now very comfortably be able to define health and what ill health is. We should be able to tell the difference between a communicable and non-communicable disease. We need to be able to define the term pathogen, which I'm very confident you guys can do, and explain different examples of viruses, bacteria, fungi, and protists. Know how pathogens cause disease and how they are transmitted, and know examples of common infections such as cholera, TB. We didn't talk about cholera ash disease, but I can mention it to you later. It's not that relevant, and we've talked about HIV and Ebola. Okay, so what this basically does is it sets the scene for what we're going to talk about next. And we've now looked at what happens when we have a pathogen coming in and what kind of pathogens can attack the body. We're now going to look at what the body does to protect itself from infections. Now, before we move on to the next topic, I think it would be wise and sensible to ask you the question of the day and be able to ask you, how confident are you with what we have covered so far? So with 5 being very confident. So remember, are you able to talk about the definitions? Are you able to identify different types of infections? Are you able to sort of slightly explain how different infections are spreading and what ways in which we can prevent the spread of certain infections? You can see in your exam questions, they seem to be talking about the common infections we have referred to, such as your salmonella and I think they're going to mention Ebola in a couple of exam questions later as well. So are you comfortable with these terms? Are you comfortable with the definitions of what a vector is and how things are spreading? So it's really nice to be able to see how you guys are sort of getting on and how confident you're finding this topic. Because what's going to happen next is, as I was explaining, the next part of the lesson is looking at how the body and what the body's defense mechanisms are in dealing with infections. Okay, so it seems to be the majority of you guys are floating around 4 and 5. Again, if it's the first time you're looking at it, obviously there can be some uncertainties, and we try to reinforce these through these interactive quizzes and the exam questions. So we are going to carry on and sort of build on what we have talked about and sort of kind of explore the immune defenses of the body now. So the immune system. What are we aiming to be able to do in the next hour or so? Now, we want to be able to describe the physical barriers and the chemical defenses of the human body that prevent infections from pathogens. So we've talked about how pathogens cause disease. We now want to talk about how the body protects us from these diseases. Understand the process of ingesting microorganisms by white blood cells. Understand the roles of inflammation and fever in fighting disease. Know that the role of the immune system is defense against disease and that it is triggered on detection of a particular pathogen. Understand that the immune system is a specific response, and you should be able to identify and be able to be comfortable with the terms antigens, antibodies, and memory cells. And know that the immune systems can retain memory, and on reinfection of the same pathogen, we can deal with it more efficiently. So again, your exam boards will have particular things they want you to know. So go over this in your sort of revision sessions. Okay. Time to look at what you guys already understand about this particular topic. So jump onto Slido again. And can you tell me what the innate immune system is? Is it a non-specific and generic response, or is it a acquired and specific response? So I've kind of thrown you guys in the deep end because I'm really picking at your brains here, because we need to be very, very comfortable with these topics and definitions. So what is the innate immune system? What do we mean by non-specific and generic response? So what's the other type of immune system? So all these things should now be ticking in your head. So just give you guys a couple of minutes, not minutes, but give you a couple of seconds to bring in your answers. Okay, So when we talk about the innate immune system, we're talking about a very non-specific and generic response. What we will see and what we will discuss is that when we talk about the immune system, there's two ways in which an immune system can respond to disease. It can respond to it in a very, very generic way, and it can respond to it in a very, very specific way. And that's what identifies and distinguishes between the two immune systems, the innate and the adaptive. Antibodies are made from which cells? So again, what are antibodies? Do you know any functions of any of these cells? Again, throwing you in the deep end because I really want to pick at your brains to see what you guys know. So what cells are antibodies made from? T cells, B cells, macrophages, red blood cells. Can you ask yourself, what is the function of these cells? Okay, so antibodies are made from B cells, and a lot of you guys are getting this right, around 48%, the higher side of you guys. So remember, macrophages is basically a type of white blood cell that's going to ingest a pathogen. Red blood cells are the oxygen carrying cells in the body. They've got nothing to do with the immune system. T cells deal with infected cells. B cells are the ones that are going to produce the antibodies. Phagocytes and natural killer cells are parts of which immune system? Is it the innate? Is it the adaptive? Again, I'm asking you guys this on the presumption that you guys know what the innate immune system is and what the adaptive immune system is. But again, this is the type of knowledge you guys are expected to know. It should be at the back of your mind. I know exactly which one's which. So phagocytes and natural killer cells are actually part of the innate immune system. So it's the whole idea that regardless of which pathogen comes in, phagocytes are going to go and ingest, natural killer cells are going to go and kill, okay? Not the adaptive immune system. Can you tell me examples of physical barriers that protect us from pathogens? So again, open floor, drop in your thingy. Wow, skin. Okay, good. What else? Nose hair. Skin. So lots of people are saying skin. What are the types of things? Skin, enzymes in the eyes, mucus in the throat. Again, excellent. What type of enzymes in your eyes? Eyelashes, good. Skin, hair, mucus, hydrochloric acid in the stomach, excellent. Tears, eyelashes, blood clots, good. Cilia, someone has mentioned cilia, fabulous. Skin, it secretes microbial, some antimicrobial substances, good. Hydrochloric acid in the stomach, that's a very, very important one. Lysozyme enzymes. So tears prevent, clean out the eye. Stomach allows pH to kill bacteria. Cilia in the lungs brush out pathogens. Fabulous. Whoever wrote that answer, excellent. Very, very comprehensive. I like the fact that you guys are saying it produces antimicrobial substances. It's very, very, it's good knowledge. Okay, so next. Plasma cells originate from which cell? Again, I'm all over the shop with these questions because you guys should be able to identify these terms from the back of your hand. So plasma cells originate from what? What are plasma cells? What is the role of T cells and B cells? All these things should be coming into mind. I'll give you guys a couple of more seconds. Interesting. Very, very interesting. Okay, so 62%. Oh, okay, we've got it changing now. Okay, fine. Now it's not so interesting. So it seems like a lot of you guys are now going for B cells, which is very true. B cells produce antibodies, and then these then turn into plasma cells that then end up turning into a antibody factory, as you will see. Okay, again, thank you very much for your responses. Any terms or definitions that you were a little bit uncertain of in those questions, no, don't worry. I'm going to be covering all of those. And the whole point of doing that is so you can identify, right, I'm not so comfortable with this. So when we go over it, you're sort of tuning into it a little bit more and sort of really identifying what parts you were not so sure about. Immunity. Immunity can be defined as the ability to defend the body against harmful foreign microorganisms such as bacterial toxins, viruses, and fungi. It's all about protecting and defending the body. The immune system is essential for responding and resisting infections and disease and keeping us healthy. Now, we were talking about two different types of immune systems. We've got the innate immune system and the adaptive immune system. Now, the innate immune system is non-specific and generic. It's a very, very rapid response, but it has no memory. So you could be affected by the same pathogen again and again and again, and your innate system would not register the fact that it's the same pathogen. It would just respond the same way all the time. On the other hand, your adaptive immune system is a bit more advanced. It's defined as an acquired and specific response to a particular pathogen. It takes a longer time to initiate the response, but it does develop memory. So when we have a reinfection with the same pathogen again, we are going to be able to initiate a faster, rapid response. An adaptive immune system is basically the hallmark of vaccines. So when we talk about the adaptive and the innate immune system, there's some important distinctions to make. Innate immune system is the first line of defense. It's not antigen specific. So again, the whole idea that it's the same response every single time, and it's a non-specific response. Your adaptive immune system is the second line of defense. So should the innate system not be so great at protecting us, the adaptive system kicks in. It's antigen specific. And again, this term we will go over in a little bit more detail when we look at the adaptive system. And the whole idea is that it's a very, very specific response being initiated. So some of the common cells you should be aware of is phagocytes and natural killer cells are going to be part of the innate immune system. B cells and T cells are going to be part of the adaptive immune system. Now, again, I'm just trying to develop you guys' knowledge here. Phagocytes are going to be involved in a process called phagocytosis, where they are engulfing and ingesting the pathogen. Your natural killer cells are going to be in the process of killing the infected cell. Your B cells and T cells, when we talk about B cells, we're going to talk about antibody production, and T cells are actually going to go and kill infected cells. Okay, so these are the sort of things. It all sort of turns into this nice little big diagram, and I don't want you to be sort of overwhelmed by it. We're going to walk through this slowly. So we're going to walk through this little flow diagram here, and we're going to begin by saying, I have an immune system. There are two parts to my immune system, the innate immune system and the adaptive immune system. We're going to take a walk through the innate immune system, and we're going to see that we've got some physical barriers that I asked you guys about, and these stop the infection entering the body. And then we've got some blood-borne mechanisms as well that basically result in the death of dangerous pathogens, and this is where phagocytes are very, very important. So let's first of all look at some definitions before I start telling you a little bit more about the innate immune system and the adaptive immune system. So your immune system is a complex network of cells, organs, and tissues that function together to fight foreign pathogens. The immune response is basically the reaction of these cells to something it does not recognize, and it's often classified as foreign. The whole concept is that we are dealing with something that is foreign, that's invaded the body, and we want to get rid of it. We have innate non-specific responses that do not target a particular pathogen or toxin. We have adaptive specific responses that do target particular pathogens and toxins. And the whole point of primary defenses is that they prevent pathogens from entering the body. And the secondary defenses kill pathogens inside the body. Now, the innate immune response. A couple of things you need to know about the innate immune response: that it's present at birth. It's known as our naturally occurring immunity. It's a very, very rapid response, and it's very, very effective against most general pathogens and harmful microorganisms. And it's so effective that sometimes the adaptive immune system doesn't even need to kick in. It provides a generic and non-specific response, so it's the same response every single time. So you know how when you go to like a hand sanitizer machine and it's going to give you hand sanitizer every single time, regardless of hand, regardless of size, regardless of color? It's the same thing here with the innate immune system. Same response each time. No memory cells are produced, which is why the body doesn't realize, oh, hang on, I've seen you before. Let me respond the same way again. It's less diverse and less potent. What that means is it's not 100% effective in protecting us against disease. So, you know, if you end up with like a cut, or you hit yourself somewhere, or you, I don't know, touch like poison ivy or something, you end up getting swelling or redness around the wound. So if I cut myself, I'm going to get some swelling and redness around the wound. And I'll have that every single time I cut myself, regardless of size, shape, matter. Okay, and that's an example of how it's the same response each time. So what we're going to do is we're going to look at the innate immune system in a little bit more detail. So ignore the adaptive and look at the innate. And I want to discuss some of the physical barriers we use to protect ourselves. And this is something you guys all mentioned, so I'm not going to be telling you anything new. Some of the ways in which our body stops infections entering our body: the skin, the mucous membranes, the eyes, ears, and the stomach and the vagina as well. And we can do things like coughing, sneezing, blood clotting, skin repair, inflammation, and fever. So let us have a look at these in a little bit more detail. Now, the first physical barrier and the biggest organ in your body is the skin. The outer layers of the skin are present with dead cells with keratin, and this actually acts like a physical barrier. Now, some of you mentioned that the skin also contains antimicrobial sort of fluids, and that also does protect us from the infection of pathogens. The next thing we can talk about is mucous membranes. Again, something you guys mentioned. Now, the mucous membranes are present at most orifices. We like to talk about the airways, and what it basically does is it traps pathogens and dirt and moves them out of the body. Now, some of you guys, again, very, very impressively mentioned the cilia. And basically cilia, I basically like to talk about cilia as the wafters. Now, if you imagine cilia sort of lining your airways, so if this is your cell and you imagine a pathogen coming in, now what the cilia basically does is it wafts, literally does this. If I showed you some like fancy technology images or videos, you would see the cilia literally just wafting. And basically what it does is it moves the pathogens and dirt out of the body, so you end up coughing it back out rather than it going into your body. Okay, and a lot of your airways, your nose, your trachea, your airways, your bronchioles and stuff are all lined with cilia, and they're very, very important. And they basically work in synchrony with your goblet cells, and the goblet cells basically produce lots of mucus, and basically the mucus will trap the pathogen and the cilia will waft it away. So a very, very two-person effort. Other barriers are things like your eyes. Now, your eyes, again, you mentioned it. You can talk about your eyelashes. Actually, I mean, how many times do we blink in a day? And when we get something in our eye, we start blinking really, really rapidly, and that's one of the protective mechanisms, and our eyelashes are there to basically protect our eyes. And the key thing here is that your eyes actually produce the fluid around your eyes basically produces lysozymes, and lysozymes are very, very important in protecting us against infections and pathogens. And we also have antibodies in our tears as well, which also helps us and protects our eyes, because eye infections can be really, really bad. Your ears, your wax actually can trap pathogens as well, and this is why they say you shouldn't use a Q-tip all the time in your ears. You need some bit of wax in there, because it's very important to help protect your inner eardrum. Some of you mentioned the stomach, and the same thing applies for the vagina as well. It's very, very acidic conditions, and they can kill pathogens straight away. If you basically ingest some pathogenic food, your stomach acid is strong enough to kill it before it gets anywhere near your intestines and really causes you a problem. Other defense mechanisms are things like coughing and sneezing. The whole purpose of coughing and sneezing is to basically expel the air carrying the pathogens. Skin repair. Now, basically, once you've cut yourself, your skin doesn't stay open forever. It may take time. Everybody has different healing times and processes, and therefore it may not happen over a day or two, but it does happen, and it's a very, very good way in which the skin protects ourselves after it's been sort of compromised from a pathogen. Inflammation is triggered in damaged tissues. You hit yourself, you're going to go red, it's going to go warm, and it's going to get swollen. And this is a way in which basically under the skin, what we have is we've got vasodilation happening, and we've got lots of blood flow flowing to the area of damage. We've got lots of phagocytes attending the scene and examining what's going on. And it's these phagocytes that are then going to initiate a sensible response, such as activating or calling for your B cells and your T cells to deal with the infection. Blood clotting. When we cut our finger with a knife in the kitchen, you don't bleed forever and fall to the floor. Your blood stops bleeding, and it's because of blood clotting. And it's a very, very good way and a very, very natural defense mechanism for plugging wounds, and it prevents blood loss. And obviously, the only reason we basically bleed is because we've cut ourselves and we've compromised the contact between the outside world and the inside world of our body. And by clotting the blood and sealing the wound, we prevent pathogen entry. And lastly, a fever. I feel like loads of people have the preconception that a fever actually causes us to become very, very poorly because it's the pathogen or the infectious disease causing organism causing the fever. However, this is not the case. Your immune system initiates the response of a fever, and your body turns up its thermostat. And higher temperatures allow the immune system to work better. So when you have a fever, it's not being caused by the pathogen, it's being caused by your immune system trying to respond to the pathogen. So, and this is just a repetition of the infographics I've just given you, and it's just I put both types of ways of information because some people prefer notes, some people prefer pictures, so this is just sort of repeating what I've said. So what we've just basically done is I've showed you all the ways, the physical barriers that our body does and has to stop infection entering. What happens if the pathogen does get into our blood? And this is when we start talking about phagocytes, okay? And these are very, very important in dealing with pathogens, and they will go through a process of phagocytosis. Now, put it simply, and again, with my awful drawings, I want you to imagine this is a phagocyte, okay? And it's going to have a nucleus, etc., but it has something called lysosomes, which contain lots and lots of enzymes. Now, what basically happens is you have a little pathogen here that the phagocyte is going to detect. And what we basically call the pros, so what basically happens is your phagocyte is going to engulf the pathogen. So it basically gobbles it up, and the pathogen is going to become internalized. So the key word here is engulfing. And what happens is the engulfed pathogen is then going to fuse with the lysosomes, and it's going to be broken down into absolute debris. Okay, and it's this entire process that's called phagocytosis. Now, to go over it in a little bit more detail. Okay, now I've sort of jumped ahead a little bit and told you about the role of phagocytes, but nonetheless, do put your thoughts down. Can you think of ways to sort of explain phagocytes in the sense of an exam style question? Do put it down again. It's about testing what you're understanding. So again, on the poll, write down what you think phagocyte, what is the role of phagocytes? Can you give any examples of any particular types of phagocytes, which I haven't told you already? And just anything about the process itself. Engulfs cells to eat pathogens. So in your exams, do not write to eat pathogens, because the correct term is to engulf. Engulf and digest microorganisms, good. Engulf and digest bacterial pathogens with their enzymes secreted from the lysosome, good. Okay, I like the fact that you guys are saying the right things, so that makes me feel like you've been listening. They engulf pathogens and fuse together with them to reduce them to nothing. Nice. They digest them. Phagocytes are cells that protect the body by ingesting harmful foreign particles, bacteria, and dead or dying cells. Good. Correct responses coming through. I should have asked you guys earlier. Identify, engulf, break down. I like that. Okay, good responses coming through. Now, which of the following cell is an example of a phagocyte? So this is what I was asking you earlier. We know the process of phagocytosis, which I could have told you about. I'm going to look at it in a bit more detail. But how do we identify what a phagocyte is? Can you identify which cell? So the correct answer is a natural killer cell. Natural killer cells are examples of phagocytes. They kill cells. So sorry, you know, when you guys' answers are coming in, it really knocks me off my thing. Okay, neutrophils are a very, very common example of a phagocyte. Okay, they're actually the most common in your body. So we've said there are, so remember, we're going to go back to our diagram again and again. We're talking about the immune system, and we're looking at the innate side of things, and we're talking about the fact that it's a very, very generic response to the infection by pathogens. Now, remember, we've said that there are physical barriers, and we've talked about the skin, we've talked about the eyes, we've talked about the nose, we've talked about the mucous membranes, we've talked about blood clotting, fevers, etc. And we've also said that if the pathogen is to come into the body, one of the ways in which we can deal with it is by digesting it and ingesting it. And this is what phagocytes do. Again, non-specific white blood cells, and they engulf and destroy pathogens and toxins. And a lot of you guys used this term, which is very, very good. Now, the other thing the phagocytes do that is very, very important is that they release chemicals to attract other immune cells. And this is basically one of the ways in which we bridge the gap between the innate and the adaptive immune system by basically releasing these cytokines, which are the fancy name for these chemicals, to tell other immune system cells you need to come here and deal with this ongoing situation. So, as I was explaining to you before, the process of a phagocyte engulfing and digesting a pathogen is known as phagocytosis. So the phagocyte, first of all, spots the pathogen. And this is basically where I was saying, here is your phagocyte and here is your pathogen. The phagocyte folds its membrane and engulfs the pathogen like so. And basically the lysosome enters the phagosome, okay? And basically then it's going to release lots and lots of digestive enzymes, and the pathogen is going to be digested into harmless products. So a more prettier picture we can see over here. To just re-emphasize the point, we've got your phagocyte here, you've got some lysosomes, and you've got the pathogen. Now, as you can see, the pathogen has been engulfed by the phagocyte to produce a phagosome. The lysosome fuses with the phagosome and releases digestive enzymes, and it basically digests it and presents the antigens onto its cell surface. And basically this antigen being presented on this is going to be a way for the adaptive immune system to come in and be like, right, so this is the antigen we're looking for, let's go and get rid of it. Okay, so that's one of the ways in which we bridge the gap between the adaptive and the innate immune system. Now, a couple of other things about other blood cells present in the body. We need to be able to identify and talk about neutrophils, lymphocytes, macrophages, and red blood cells. And without overly dropping too much information on you, allow me to sort of simplify it a little bit. Your red blood cells are the ones that carry the oxygen. You should know that your lymphocytes, when we talk about lymphocytes, you need to think of two letters, your T cells and your B cells. And you should know that these are part of your adaptive immune system. So this is how your mind should be thinking about it. Your neutrophils and your macrophages are important in phagocytosis, and they are part of the innate immune system. Okay, now to go over them in a little bit more detail. Now your neutrophils are basically the most common phagocyte with multi-lobe nucleus. So remember, in normal cells we will have like this boring looking nucleus. Our neutrophils are a little bit extra and they have this multi-lobe nucleus. They are made in the bone marrow and live for a very, very short amount of time. And they contain many, many lysosomes. So again, one of the reasons why that's very, very important, if it's a phagocyte, we need loads of lysosomes there to be able to digest the pathogen. So it's a very, very good adaptive mechanism. And basically what happens is when you injure yourself, we release these chemicals into the body called cytokines. And the neutrophils basically move to the area where there's loads of cytokines being released by the site of infection to go and deal with what's going on. And again, we refer to lysosomes a lot. And what you need to remember is that lysosomes basically contain lots of hydrolytic enzymes. And hydrolytic basically means is they enzymes that are very good at breaking things down. And remember, we talked about how when we ingest that pathogen, it's going to then fuse with a lysosome and it's going to break it down into absolute debris and harmless products. A macrophage is a large cell with a kidney-shaped nucleus. They basically travel in the blood as monocytes until they settle in body tissue. Their role involves initiating the immune response, and they act as antigen-presenting cells post-phagocytosis. So where neutrophils are just consuming and getting rid of, the macrophages are very important in presenting the antigen they just consumed to the rest of the immune system. So macrophages basically actually walk around the blood as monocytes, okay? And they can turn into, now this cell can differentiate into a dendritic cell, it can differentiate into a macrophage, and it can differentiate into an osteoclast. You don't need to know so much about the osteoclast and the dendritic cells, but I was just using this diagram to emphasize the idea that it's coming from a stem cell, kind of like undifferentiated cell such as a monocyte, and it can turn into a macrophage. And the macrophage, remember, is going to be responsible for phagocytosis and antigen presentation, which I will tell you about in a little while. And this, I thought this was a really cool picture about that's basically the phagocyte, and this is ingesting the pathogen. It's literally gobbling it up. Right, I've talked a lot about antigen presentation, and I'd like you to take a look at this little diagram here. Now, can you see we've talked a lot about your phagocytosis process. Now, remember we said that the macrophage takes up the antigen by phagocytosis, and we've said that the macrophage breaks down the antigen into fragments in the lysosome. Now, what happens is a very fancy receptor is basically then going to present that antigen to the outside of the macrophage. And what happens is it's then going to present this protein to the T cell. Now, what the T cell can then do is go and identify this pathogen anywhere else and go and kill it. It can identify the pathogen in any other cell and go and kill it. Okay, so it's the whole idea that how does the adaptive immune system even realize what's going on? It's because of this little antigen presentation process. Okay, and the key take-home message here is that the macrophages link up the immune system with the adaptive immune system. Now, remember I said lymphocytes in my little summary were B cells and T cells. And when we talk about the immune system or blood cells, we can talk about your red blood cells, we can talk about your neutrophils and your platelets. The ones we are interested in, and when we talk about lymphocytes, is all these ones. Okay, your B lymphocytes, your T lymphocytes, and your natural killer cells are also an example of it. Okay, and lymphocyte is another way of saying B cells or T cells. You can use it synonymously. Red blood cells are pink in color with a biconcave disc, and they are all to do with the oxygen carrying capacity of the body. So that's all you need to know about this. So again, to summarize, I'm very big on summaries because sometimes I can feel like it feels like information overload. Neutrophils are a type of phagocyte, and they release cytokines, and they are going to go and just simply ingest pathogens. Lymphocytes are T cells and B cells, and these are part of your adaptive immune system. And macrophages are involved in phagocytosis. Remember, they walk around the blood as monocytes until they have to go and deal with an infection. So then they become macrophages. And the key thing here is, like neutrophils, they go through phagocytosis, but then the key thing they do is they end up doing antigen presentation to the adaptive immune system. So now I have a couple of questions coming up for you about what we've just sort of covered. Which cell is the most common phagocyte? Again, this just gives you a minute to sort of process all the information I've bombarded you with. So which cell is the most common type of phagocyte? So again, how well were you listening to what I was saying? Is it a basophil? Is it a neutrophil? Is it a dendritic cell? Is it a macrophage? What are we going for? And try to think about what the individual functions are of these different cells that I'm telling you about. Let's give you guys a couple of seconds. So, which cell is the most common phagocyte? It is your neutrophil. Okay, you need to remember that. And a lot of you guys are saying neutrophil, so very well done. Now, which cell will present the antigen to the T cells? So again, it's about knowing the individual cells and whether you can really distinguish the roles from one another. So I'll just give you a couple of seconds. So which cells will present the antigen to the T cells? So majority of you guys are saying macrophages, and that is very, very true and correct. So well done. Macrophages are the ones that, like neutrophils, undergo phagocytosis, but they go one step further to present the antigen they've just discovered to the T cells. So again, well done for those of you who are signing in with your names. It seems like some of you guys are doing very, very well, and it's nice to see sort of the interaction and to see how you guys are sort of going with these questions. Okay, another question. I keep saying exam questions. Which structure in phagocytes helps digest pathogens? So again, this is something I feel like I said like maybe 6 times across 6 different slides with my fancy drawings. So which structure in phagocytes helps digest pathogens? Is it the Golgi body? Is it the lysosomes? Is it mitochondria? Is it my receptors? What are we going for? A couple of more seconds. Lysosomes. 92% of you guys are very correctly identified that it's the lysosomes in the phagocytes that help digest pathogens. Fabulous. Remember, they contain lots of hydrolytic enzymes that help break it down. So, name 2 types of lymphocyte cells. Again, something I've told you about, because sort of going to leave it as an open B and T cells, and it's nice to see whether you guys are going to say the right thing or the wrong thing without being prompted with multiple choices. T cells and B cells. Good. T cells and B cells. Lymphocytes, good. So whoever's saying phagocytes, please don't use the term phagocytes, simply because phagocytes are not a type of lymphocytic cell. When we talk about phagocytes, we are talking about neutrophils. We are talking about macrophages. It's the T and B cells we want to be talking about. Good. Someone has also gone one step further to say natural killer cells. Fabulous. Okay. Next question. What is the role of T cells? Again, I'm opening the floor and kind of letting you guys tell me what you think the role of T cells is. To locate the pathogens, engulf them. To kill pathogens and infected cells. Some of you guys have some good responses. So immune system to respond to antigens, kill the pathogen, directly kill host cells. Is it going to go around killing any host cell, or is it going to kill a host cell that's infected? To find the pathogens, possibly to engulf and kill pathogens. Someone's saying create antibodies. So if I could flag that as a complete no-no, that's the type. It's the B. Okay, I'm not going to say it in case that's the next question. To find pathogens. Okay, so we've got a couple of mixed responses coming in, but the main one people are saying is to kill pathogens and identify cells that have pathogens in them to then destroy the cell. What is the role of B cells? So let's jump the queue and go to the other lymphocyte. What is the role of B cells? What do you know about B cells? To make antibodies, to release antibodies. Produce antibodies. Good. So a lot of you guys are coming up with the right thing. To engulf and kill pathogens. Not so much. It's the antibodies we are talking about. They're producing antibodies. Good. I'm seeing they're involved in the humoral response. So anyone who said that, well done. Yep, they produce antibodies. Okay, good. So a lot of similar answers coming in. I feel like you guys are more confidently expressing and explaining the role of B cells to me as opposed to the role of T cells. And again, this is something we will differentiate as we go through more content. So when we talk about our big fancy diagram here, again, to put things into context, we've discussed what the immune system is. We've looked at the innate immune system, and we've talked about the different physical barriers that are present to help stop infection entering the body. We talked about the role of phagocytes and how they ingest dangerous pathogens. We are now talking about the adaptive immune system, and I've just introduced the concept of T cells and B cells to you. And now we're going to look at what role they play in protecting us against infection. And this is just me being a little bit extra and saying, you guys should all be happy with the innate immune system. We're now going to move on to the adaptive immune system. So your adaptive immune system, if I could show you one picture, and if it was one thing you wanted you to take away from the adaptive system, it's going to be this. So just follow my cursor. When we talk about the adaptive immune system, we're talking about your B cells and we're talking about your T cells. Now, your B cells are basically going to have all these fancy antibodies stuck onto them. These antibodies are going to bind to a particular specific antigen, okay? And what that's basically going to do is, once we've identified which antibody is binding to which antigen, that's going to stimulate further production of that particular antibody. And when this happens, our B cell transforms into a plasma cell and it turns into an antibody creating factory. When we have a T cell, okay, the T cell is basically responsible for identifying the antigen. And the T cell is a little bit more of a leader, and it then goes and delegates other roles to other T cells. Once the T cell has an antigen in place, it can then basically turn into a cytotoxic T cell, which then goes and kills any cell that's infected. A helper T cell, and what we will find is that the helper T cell actually goes and helps your B cells turn into plasma cells. And the suppressor T cell is very, very important to basically control our immune response. And it's kind of like a negative feedback system, you could say, where it basically then tells the T cell itself, right, okay, I think you've killed everything you needed to, you need to stop now. And this is basically the adaptive immune response in a nutshell. Again, I'm going to go over these things in a little bit more detail, but it's nice to sort of have an overview. Your adaptive immune response requires immune cells and proteins within the blood to attack, destroy, and eliminate foreign pathogens. It's a slow response. It's a delayed response because the immune system cells need to develop antibodies to one specific pathogen, so it takes time to kick in. But the good thing is it may be slow and steady, but slow and steadies win the race. It develops memory. And once the pathogen has been cleared, should we ever be reinfected with this pathogen, we don't have a problem in dealing with it. Okay, the next immune response is going to be quick because we've established immunological memory. Now, the adaptive immune system does a very, very important thing, and it basically distinguishes between self and non-self cells by using antigens. Now, every cell has various proteins and molecules on its cell surface, and these are called antigens. And they act as a recognition device to distinguish between self and non-self. It's the adaptive immune system which will be activated by antigens of a foreign pathogen. Now hold on to this for a second because I'm going to show you this in a very, very creative way. Now, what do we mean by self and non-self? I want you to imagine a couple of different cells, a green cell, a red cell, a blue cell, and a baby blue cell. These are cells that are part of our own immune system, okay? And we are talking about antigens that are proteins on top of these cells that allow us to recognize that, okay, this is a body protein. So the green cell has a star-shaped antigen, the blue cell has a triangle-shaped antigen, this one has a smiley antigen, and this one has a heart-shaped antigen. Now, Your immune system cells, such as your B cell or your T cell, has this already fixed into their hardware system, that the heart-shaped antigen, the star-shaped antigen, the triangle-shaped antigen, and the smiley antigen are all part of the body. They are self. They're part of us. They're part of the team. They're part of the group. Okay, now, when we have a cell that has an antigen that's not part of our own hardwired motherboard or memory card, we start thinking, no, we need to deal with it. And then the immune system goes all hammer on it and it's like, right, we need to get rid of you. I hope that explains the concept of antigens, okay? It's the whole idea that we will recognize the antigens from our own body cells as our own, and antigens we don't recognize, they are non-self and they need to be eliminated. So, all cells and viruses have antigens present. As I was explaining, the antigens are proteins that stick out of membranes. The body can recognize these antigens as self or foreign, and these foreign antigens can be classified as pathogens, other cells, or abnormal or damaged body cells. So even cells from other organ donations and transplants can be identified as non-self. And it's this identification system that allows us to say, okay, I'm going to get rid of you, but I'm going to keep you because you're part of my team, but you're not part of my team. And then what basically happens is the foreign cell is going to be labeled and then destroyed. So one of the ways in which the antigens can be dealt with is, so imagine this is a bacterial cell, and we have now recognized that this is a bacterial cell with an antigen that we don't like. What happens is your antibodies are going to go and bind to the antigen, okay? And what you need to remember is that every antibody is going to bind a very, very specific antigen. So this antibody will bind a square antigen, or this antibody will bind a circle antigen. They're all very, very specific. And as you will remember, it's the B cells that produce your antibodies. So I've already introduced the terms lymphocytes to you, and they're defined as white blood cells. They're involved in the specific immune response. Again, no new information there. Two types, your T cells and your B cells. And the key distinction to make, and I really like this little diagram I found off the internet, it basically says B cells attack invaders outside the cell, and the T cells attack infected cells. Okay? These are the ones that are making the antibodies, and these are the ones binding the antigen and recognizing the antigen. So again, going back to our main graph, what are we doing so far? We are looking at the immune system, we're looking at the adaptive immune system, and we're talking about B cell immunity. Before I tell you anything else, the first thing that should come to mind when we talk about B cells is antibodies, okay? You also should start thinking about antibody factories, which are basically your plasma cells. And if you sort of note this over here, when we talk about B cell immunity, we can refer to it as humoral immunity. Now, what happens is we will have a B cell, and it's going to recognize your antigen, okay? And then it's going to say, right, I need to turn into an antigen producing factory. So it's basically going to turn into a plasma cell. And then the antibodies are going to be released. And then we're going to create these antibody antigen complexes to deal with the pathogen. Okay, so B cells, B for B, they mature in the, this is an awful drawing of the bone, but they mature in the bone marrow. They are covered in antibodies. So again, what I'm basically saying is that one particular B cell is going to have 10, maybe 10 different types of antibodies present. And basically what happens is when one specific antigen binds to that specific antibody, it's this antibody that's going to be produced, not all the others. So this is what we mean by clonal selection. A specific antigen will bind to a specific antibody. And then we have the expansion where we basically internalize the antibody and replicate it. So what happens is the B cells are then specialized into plasma cells, and these are the ones that create the antibodies, and then later we have B memory cells that then float around in the blood forever to then deal with a reinfection should it happen. So again, we have a B cell. It's going to bind one of its antibodies to an antigen. It's going to turn into a plasma cell that's going to then make and release antibodies. And it's also going to create some B memory cells that will remain in the blood to deal with another infection from the same antigen in the future. And this is also known as the humoral response, otherwise known as humoral immunity. So again, just re-emphasizing, the whole concept of humoral immunity is that you've got your T cell covered in antibodies. The antibody is going to bind a particular antigen, and it's going to create plasma cells that are going to produce lots of antibodies for us to deal with this infection. Now, earlier on, I basically talked about how this type of immunity prevents us from reinfection and allows us to deal with the second response very quickly. So can you see over here, when we have an initial antigen exposure, our primary response actually takes around 4 weeks to initiate. Okay, or so not 4 weeks, but 2 weeks to actually initiate and build antibodies for, because we're just processing it. Where we have a second antigen exposure from the same antigen, the rapid production of antibodies is very, very good because we've seen it before. And it's this whole humoral immunity concept, and it's this concept that's basically implemented in vaccines as well. The whole idea is that you see the pathogen once, so when you see it again, you're not going to be so badly infected by it. So just a little bit about what antibodies are. They're part of this specific immune response. They're made from B cells, very large proteins. Now, key thing to distinguish is, remember, the B cell itself is just identifying the antigen. It's the plasma cells, which are the antibody making factory, that are going to secrete the antibodies. So you need to remember the distinguish factor between plasma cells and B cells. They are complementary to a particular antigen. And what do? They basically make the antigen harmless, and they can start the process of phagocytosis. You don't need to know antibodies in too much detail, but the whole concept here is that we have a variable region and we have a constant region. Okay, and it's the variable region that can change, and it gives them the ability to bind to different antigens. They're Y-shaped molecules made of polypeptide chains, so they're basically a protein, and they're held together by little disulfide bonds here, and there's your little hinge region for flexibility. So again, just some things I want you to bear in mind here. Now, one of the ways in which antibodies work to deal with infections is by basically something called neutralization. Now, what happens is it can bind to all the antigens, and the pathogen can no longer then enter the cell. It then tells the phagocytes to come to it and to basically ingest and digest the pathogen. So one of the things it's basically doing is it's binding to the pathogen, holding it there until the phagocytes come through for phagocytosis. It also, what it does is it basically, several antibodies can clump together with pathogens and then wait for the phagocytes to come to them to then ingest the pathogen. So it's basically a big broom. The antibodies act like a big broom that collect all the pathogens, and then they take them to phagocytes to deal with. Okay. Now, again, a nice little summary diagram. The whole purpose of B cells is to initiate the humoral response. The B cell is going to recognize a foreign antigen. It's going to replicate and realize, right, I need to turn into a plasma cell to replicate this particular antibody. Remember, I told you earlier that it's the T helper cells that will help the B cell turn into a plasma cell, which then goes on to secrete antibodies, and then they go on to destroy the pathogen in various ways. Now, I'm going to stop talking there, simply because I am very well aware that we are heading up to 12:30. We are more than halfway through our content. We're going to stop at the discussion of B cells, and after lunch, we're going to pick up the discussion about T cells, and how they are different to the B cell response. And then we will go on to do some exam questions and then move on to the other topics. It is a very, very content-heavy session, and I am trying my best to make it as interactive as possible. I think it's a very, very sensible time to say pause and sort of start moving towards our lunch break. As you guys are aware, we are doing lunch break from 12:30 to 1:30. So take a minute, give yourselves a break, go on and enjoy your lunch. And well done for really interacting through the Slido polls. It's nice to see you guys are putting in your answers, and it's a good way to sort of be able to tell how you guys are able to distinguish between different terms I'm talking to you about. So I do really, really encourage participation in the polls and the sort of quizzes we sort of put out. So again, it is lunchtime now. We're going to go, as you know, lunch is going to be from 12:30 to 1:30. Go and enjoy your lunch break, and I will see you guys in exactly an hour. I do hope you guys found this morning session useful. After lunch, we're going to sort of go over the rest of T cells and B cells, and then we'll do some exam questions and then move on to the next couple of topics. So I will see you guys in an hour. Okay, guys, welcome back to the afternoon session with Elite Medics Online. We're sort of covering Year 11 biology content today. I hope you guys have had a very restful lunch break, and you guys are sort of all ready to go for the afternoon session. Now, just before we broke up for the afternoon session, we had talked an awful lot about immunity and the immune system, and we had talked about the adaptive immune system, and we had just come on to the we'd covered the innate immune system and we were doing the adaptive immune system, and we were talking about how B cells work. Now, just to sort of emphasize what we were talking about, what we're going to do is we're just going to go over to B cells and then T cells, and then we'll move on to some exam questions. So just before we broke up for lunch, I'm just going to check if everything is sort of okay on my side in terms of screen. Okay. So just before we broke up for lunch, we talked about humoral immunity, and we talked about the humoral response, which was initiated and directed by B cells. We said that the B cell will recognize a foreign antigen. It would then divide into a plasma cell, which would then become a antibody producing factory, secrete antibodies, and then go on to destroy the pathogen. We also made a point that our T helper cells, which is a type of T cell, would help in this process. Now, to summarize, we should know that the plasma cells are the B effector cells, and they're the one that secrete the antibodies, and it's the memory B cells that are produced when exposed to a foreign pathogen, and they will remain for years and years searching for the same antigen. And this is why when we are reinfected, our secondary response is much faster to the same pathogen. Now, what you have to remember is that plasma cells and T cells themselves actually only live for a few days because they're part of the initial response, but it's the memory cells and the T memory cells that are going to survive forever. So the T cells, on the other hand, can actually differentiate and go on to kill infected cells, and that's their main role. Where the bone, remember we said B cells mature in the bone marrow, your T cells actually mature in the thymus gland, okay, which is the little redundant gland sitting in your neck. It's covered in antigen receptors, and it only responds to antigens presented on body cells. So remember when we talked about your macrophages, and we said that it was the macrophages that were going to present the antigen, and it's this which is then going to be picked up by your T cells. Okay, so what basically then happens is your T cells, once they pick up that pathogen or antigen, they're then going to either become a T killer cell or T helper cell or a T memory cell. Now what happens is the receptors bind to the antigen, and we have this T cell dividing into a specialized type of T cell. It's either going to divide into a T killer cell, it's either going to divide into a T helper cell or a T memory cell, and depending on what it's divided into will determine its function. As you can see over here, already being highlighted in this diagram, if it's a T killer cell, it's attacking infected cells. If it's a T memory cell or a T helper cell, it's going to stimulate the B cells to divide, and the T memory cell will continue to scan the body's blood for reinfection. And this is associated with cell-mediated immunity. So where your B cells induce humoral immunity based on antibodies, your T cells are associated with cell-mediated immunity. Okay? So again, it's the T cells differentiating into memory cells, T helper cells, or T killer cells, and they are leading to the death of the infected or damaged body cell. Now again, T cells are produced in response to invading pathogens, and this is just repeating what I've just said in the first sort of slide there. And the key thing here is once the T cell, so remember I told you the key thing is the macrophage is presenting the antigen, it's phagocytosed to the T cell, and then the T cell is then going to grow appropriately. So phagocytosis occurs, the phagocyte presents the foreign antigen to a T cell, And when it's a complementary antigen to the T cell's receptor, it becomes activated. And once the T cell is activated, it then replicates. And it then replicates into the following type of T cells. T killer cells, which will destroy the pathogen infected. Now, your T helper cells actually stimulate B cells and T cells to further divide, and it helps drive the specific immune response. It releases chemical messengers that go on to activate B cells as well and turns the B cells into plasma cells to secrete the antibodies. The T regulatory cells or the T suppressor cells basically are all about, I like to think about it as negative feedback. They are the complete opposite to the helper cells. Where the T helper cells are stimulating an immune response, the T suppressor cells are inhibiting the T cell immune response, and it's ensuring that the immune response does not go into overdrive in response to the pathogen. And then the whole point of adaptive immunity is to generate some form of memory. It's the T memory cells that will recognize and remember the antigen for future encounters. So that sort of brings us on to the end of the discussion about your B cells and T cells, and I really hope I've been able to emphasize the key points to you guys. So let's have a go at some questions. I was going to say infections. Let's have a go at some of the questions I have for you. So again, just as before, you just need to jump onto slido.com and type in the code to enter the polls, and we will then go through some of these answers. So the question here is, which cell releases antibodies on initial exposure to the antigen? So again, really, really important to differentiate between the different functions of these cells. I'll give you guys a couple of seconds to pop your answers in. So which cell releases antibodies on initial exposure to the antigen? Really important to be able to differentiate between your T cells and your B cells. So remember, it's the plasma cells. Now, a lot of you have said B cells. The role B cells plays is to identify the antigen on its antibody. It then differentiates into a plasma cell, which then produces the antibodies needed and then secretes the antibodies into the blood. So you have to remember it's the plasma cells doing it. B cells are responsible for recognition. Which cell stimulates antibody production in B cells or facilitates antibody production in B cells? So again, have a very, very close look at the statement being made. Okay, let's give you guys a couple of more seconds to put some answers through so everybody has a turn. Okay. T helper cells, fabulous, so good. So remember, T cells can differentiate into different things. We have T helper cells, T memory cells as well. And it's the T helper cells that sort of give the B cells a little bit of a push to sort of go on and, you know, go make some more antibodies. T cells mature in the thymus, the bone marrow, the thyroid gland, or the lymph nodes. Where do our T cells mature? And on the reverse side, I want you to think about where B cells mature as well, please. Couple of more seconds. Okay, so the T cells mature in the thymus. Again, it's a redundant gland sitting in your throat. The B cells are the ones that are going to mature in the bone marrow. B for bone marrow, B for B cells, T for thymus, T for T cells. That's how I like to remember it. Humoral immunity is initiated by which cell? So again, testing to see whether you understand these terms. So humoral immunity is initiated by your B cells. Remember, we talk about the humoral response. We're talking about the production of antibodies, and it's your B cells that are involved in this. T cells are involved in cell-mediated immunity. Which cells destroys infected body cells? Again, very, very simple questions. Testing key important facts. Again, kind of hoping for 90%, 95% of you guys getting it right. Good. Oh, 85. Good. So remember, it's the T cells that destroy the infected body cells. They're able to recognize antigens being presented to them by the antigen presenting cells, which are otherwise known as the macrophages. And then it's the B cells that are going to go around producing the antibodies. Okay. So that brings us on to the end of the discussion about the two different types of immune systems we have. So again, we've looked at the innate response, which was non-specific, and we've looked at the adaptive response, which is more specific. With the adaptive response, we've got the B cell and we've got the T cells, and it's the B cells that are going to produce antibodies via the plasma cells, and they will hold some form of immune memory via B memory cells. And the T cells can differentiate into T killer cells, T helper cells, and T memory cells to first of all deal with the pending infection and then initiate some form of immune memory. So again, something I alluded to earlier, remember the whole point of generating some form of immunity is that should we ever come across the infection again, we are able to deal with it quickly and strongly, which is what the whole concept of vaccines is as well. So remember, when we have a primary response to an infection, there's no memory cells. It takes a few days to produce antibodies, and the memory cells are then produced during it. When we have a second infection, we have memory cells that are going to respond very, very quickly, and the amount of antibodies produced is very, very large as well, because we've already seen what we need to do. So recall a time where, you know, when we talked about your T helper cells, no, sorry, your T suppressor cells or your T regulatory cells. Now, your T regulatory cells we've mentioned were very important for, if I can just go back, very important in actually suppressing the T cell immune response to make sure everything was shut down properly. Now you can see every single cell here has a very, very important role to play. Now, if any of this cell differentiation or expansion of cells is to go amiss and go bad, we can end up with certain cancers. Lymphoma is actually a type of cancer of the blood, and it develops from your white blood cells. And basically it happens when your lymphocytes change and grow out of control, and you end up having too many of these and you end up becoming very, very poorly. Some of the treatments for lymphomas are things like chemotherapy, radiation therapy, and you end up being very, very sick. You'll have lymph node enlargement, your kidneys will start going bad, bone marrow becomes weak, your bones and your joints hurt, spleen becomes enlarged trying to get rid of all the blood cells, you become weak, you lose weight, and you have fevers and night sweats very regularly because your immune system is compromised. It's a very important condition to know about. And this is an example of a very rare type of lymphoma called Burkitt's lymphoma. And you can sort of see how it's really, really affected this child here. And there's so many different types of lymphomas. We could turn it into a whole lecture series, but this is just an example of how things can go really bad when a type of blood cell starts growing out of control. Autoimmune diseases is something we need to touch on when we talk about the immune system. And while the immune system is great as dealing with foreign pathogens, where we have some form of miscommunication between the immune system and our own proteins, we end up with autoimmune disease, where the body's immune system attacks its own cells. One type of autoimmune disease is something like systemic lupus erythematosus. This is basically where we have autoimmune condition where we end up attacking our own certain types of blood vessels, and it leads to this massive immune system inflammation where the patient actually presents with this very distinct butterfly rash when they're exposed to sun, and they end up with a kidney condition where they have lots of blood and protein in their urine. They'll end up with heart conditions as well, and they become quite poorly. And this is because of the immune system going into overdrive and killing off cells with certain proteins on it. Another condition is rheumatoid arthritis, which basically is where the immune system attacks the patient's own joints. So here on this side, we have a healthy joint, and here we have a very, very rheumatoid joint where it's been attacked and there's lots of inflammatory cells present. And for some reason, the immune system refuses to recognize the joint as its own, and it goes on to destroy it. And you can see how we end up with these deformities with patients with rheumatoid arthritis. Diabetes mellitus type 1 is also an example of a autoimmune condition. To summarize, as we have talked about on and on, the body has a primary and secondary response to pathogens. The body has innate non-specific response and an adaptive specific response to pathogens. The primary defenses include skin, mucous membranes, wax, and acidic environments. There are two types of phagocytes, both are non-specific responses. Phagocytes engulf and digest pathogens and toxins. Remember we talked about the two types of lymphocytes, which are your T cells and your B cells. They go through clonal selection, whereby their receptors match up to an antibody, and then they go through clonal expansion, whereby T cells become either T killer cells, helper cells, or memory cells, or B cells become plasma cells. Okay? Now, cell-mediated immunity is associated with T cells, where it's responding to an antigen on the body cells and dealing with infected body cells. Humoral immunity is associated with antibodies. The antibodies are proteins produced by the immune system, and they have a constant and variable region. Different antibodies will bind different antigens. And the variable one is complementary to one type of antigen. So what we're going to do is we're going to try and attempt some exam questions with this topic, and hopefully that will reinforce some of the things we have discussed. So the parts of the blood can be separated from each other by spinning the blood in a centrifuge. And it says the below image shows the separated parts of a 10 centimeter blood sample. So what we have is we've got plasma, we've got white blood cells and platelets, and it says calculate the percentage of the blood that is made of plasma. So again, it's just about reading this. So if we have said it's 10 centimeter cubed of blood, and if we assume this is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, then the plasma is around 55% of the total, because what we're basically reading is it's the idea that we're assuming if this is 10 and it's around here, we're looking at sort of 55, 56. So what we basically do is we're doing 5.5 divided by 10, which is the total, multiplying it by 100 to give us the percentage. So really take a minute to look at this scale on the side. Now it says name three chemicals transported by the plasma. Anything in the body can be transported by the plasma. We can look at proteins, we can look at hormones, we can look at antibodies, enzymes, water, vitamin. I mean, your mark scheme gives you loads, and I think this is really nice, 3 easy marks. So amino acids, antibodies, carbon dioxide, enzymes, glucose, proteins, vitamins, you know, the common ones. So again, 5 very, very simple marks, 2 for reading a scale, and 3 for naming very simple substances in your blood. Now, just to sort of put it into context, when we talk about your blood, we basically have your red blood cells, we have your white blood cells, and then basically they all float around in this liquid called the plasma, and it's the plasma that's then going to carry all of these other things, such as your platelets, your antibodies, your amino acids, etc. Okay. Now it says, in this question, you will be assessed on using good English, organizing information clearly, and using specialist terms where appropriate. Now, white blood cells are part of the immune system. White blood cells help the body to defend itself against pathogens. Describe how pathogens cause infections and describe how the immune system defends the body against these pathogens. So first of all, remember, pathogens cause infections by releasing toxins, right? And some of the ways in which the immune system defends the body against these pathogens is basically by your phagocytes. So basically these will go and ingest the pathogen. You can then go on to say that the phagocytes will then present the pathogens to your B cells, which will go on to produce antibodies, and it will present it to your T cells, which will go on to kill the infected cells. You can then go on to say the B cells and the T cells can initiate some form of immune memory that will then protect you from further infection. Okay, so all the spiel we've been talking about previously about what the toxins do, what phagocytes do, what B cells do, T cells, will award you these 6 marks. So having a look at what the mark scheme is looking for in particular, what you should be able to basically say is some of these points. So viruses and bacteria are pathogens. They reproduce inside the body and they produce toxins that make us feel ill. Now, your white blood cells can ingest the pathogens. So this is when we're talking about your phagocytosis. They can destroy the pathogens by producing antibodies. They destroy particular pathogens. So what I would basically say is, when we're talking about they destroy particular pathogens, this can be done by your T cells, and producing the antibodies is a function of the B cells. And we can also produce some antitoxins as well. And also it says here, credit memory cells that then leads to immunity from that pathogen. So basically giving a nice comprehensive overview of phagocytes, T cells and B cells, and the fact that this can lead to immune memory should award you the 6 marks. A very nice way to revise over what we've been discussing. Now it says, give 2 ways in which white blood cells protect us from disease. They produce antibodies, they produce antitoxins. They can also help us with the ingestion of pathogens via phagocytosis. Now it says, explain as fully as you can how immunization protects us from disease. So remember, immunizations, vaccines, have a weak form of the pathogen. They basically infect the patient with a weak form of the pathogen. The patient then responds from its B cells and produces antibodies. Okay, now these B cells will stay in the body as memory cells, and they will know that this particular antigen was there and we produce these particular antibodies to it. So then what happens is, when we have reinfection, the body has this massive store of antibodies to deal with this infection and prevents us from getting super ill. Okay, so that's the concept behind immunizations. So for 5AI, you can say anything like ingesting microbes, producing antibodies, and producing antitoxins, and this will get you your marks. For part AII, remember, it's the whole idea of injection of weak microbes stimulates antibody production, and these can be produced quickly on new infection. Okay, so it's these simple parts. In fact, What they're looking for in the mark scheme seems to be a lot more simpler than what I've just explained to you, but this is a nice sort of points to memorize to help to make sure you really gain all those marks when you're doing this type of exam question again. So as you can see, the type of questions you're getting for this particular topic are quite straightforward, and it's very important to therefore know exactly what you're talking about in terms of your immune system and the different types of immunity present to acquire all these exam marks. Okay, so again, just now what you guys should be able to very comfortably talk about is the physical barriers and the chemical defenses that the human body has to protect us against infection. Know about the different white blood cells and how we use the immune system and how it's triggered on the detection of a pathogen. We should be able to define the terms antigens, antibodies, and memory cells, and also talk about how immune memory is made from these mechanisms. Now, the next thing we're going to talk about is a little bit more technology based, and it's less biology, but it's very important aspect of science, where we talk about medications and we talk about the development of drugs. What we want to talk about is and understand what a vaccine does and what purpose it serves. What is the advantages and disadvantages of immunizations, including the concept of herd immunity? Why antibiotics kill bacteria, but they can't kill viruses. We want to understand what a monoclonal antibody is and know that the uses of a monoclonal antibody are and what the advantages and disadvantages are. And we're going to discuss how we discover drugs and put them out onto our shelves in our pharmacies. So my question to you guys, what is a monoclonal antibody? What can you tell me about monoclonal antibodies? So again, type away your answers. Do you know anything about the process, what they're used for? I'll take any suggestions. So any keywords you may know of. Okay, so what do we have here? A drug that contains antibodies split to a pathogen to treat disease. They are used in pregnancy tests. It is simply an antibody made by cloning, so that's a very good one. They're genetically engineered. Antibodies produced from lots of clones of a single white blood cell. Someone has said hybridoma. Good. Single specific antibody cloned. A monoclonal antibody is a lab-made antibody using rats. Good. So they can reproduce and have lymphocytes bind to it and thus allowing for reproduction. Okay, I'm seeing some very key points come through. Good. There are antibodies which are produced by lymphocytes. Use B lymphocytes from mice and fuse them with a myeloma cell. Okay. Binds to HCG. A lot of you are talking about pregnancy tests. A lymphocyte is fused with the tumor cell and it creates a hybridoma. The test drugs on animals such as mice. Presence for a specific antigen. Good. They can reproduce and have lymphocytes bind to it and thus allow for reproduction. Okay, some good suggestions coming through. We'll discuss this in just a little while. Antibiotics will harm the host organism they are using to be treated. So this is asking you about how do antibiotics work? Do they harm the host organism? What is the deal with them? Is this true or is this false? Okay, So this is very much false. Antibiotics do not harm the host organism. They are designed to harm the bacteria they are dealing with. Antibiotics are used to treat which infection? I feel like I kind of accidentally gave away the answer there. So I'm expecting 100% considering you guys are listening to me. So, and this is again a very, very important distinction to be making. What infections are antibiotics used to treat? Give you guys a couple of seconds. Okay, so antibiotics should be identified as being used to treat bacterial infections. We cannot treat viral infections with antibiotics whatsoever. Which scientist discovered penicillin? This is just for a sort of additional question because it comes up sometimes, and I think it's good knowledge to have when we talk about antibiotics. You can't talk about antibiotics without talking about this particular scientist. So the correct person is Alexander Fleming. He was the one who accidentally discovered penicillin. Mendel is all to do with genetics and inheritance patterns. Vaccines contain active pathogens. Is this true or is this false? So this is false. Vaccines will contain dead pathogens or ineffective pathogens or inactive pathogens because we don't want to overwhelm the immune system. Okay, so let's talk a little bit about antibiotics and vaccinations. Antibiotics work to kill or damage bacteria. B for bacteria. They are ineffective against viruses. One of the ways in which antibiotics are really good is because they deal with the actual bacteria itself. The thing with viruses, remember we said very, very earlier on in the day that viruses actually infect the host cell, and our antibiotics aren't designed to damage host cells, so it will never clear out the viral infection. Two types of antibiotics. We have antibiotics that can prevent growth of bacteria, and we have antibiotics that kill the bacteria. One of the most common antibiotics you should be aware of is penicillin, and it actually works by weakening the bacterial cell walls, and it leads to the cell bursting open, okay, and renders it harmless. Antibiotic resistance is a very, very important concept to be aware of, and it's a growing problem in modern medicine. If you imagine 6, 7 sort of bacterias, I'm just going to bring my cursor up, right? And one of them's kind of resistant. You take antibiotics, you don't complete the course, or something goes wrong, and you don't end up clearing all your bacteria out. All the non-resistant bacteria will die, but the resistant bacteria stays alive. And what it does is it's now not only knows what antibiotics and how to deal with it, it can then go on to reproduce. So if the patient still is unwell and has an infection, what antibiotics are we supposed to give them when this now strain is very, very resistant? And this is becoming quite a common problem. The overuse of antibiotics has caused antibiotic resistance. Random mutations often occur that make the bacteria resistant. And the antibiotic creates a selection pressure, so you end up clearing out all the weak ones that don't have this fancy mutation, and the resistant strain reproduces, and we end up with a superbug that can now no longer be treated with antibiotics, such as that common example is MRSA, which is methicillin-resistant staph aureus, and it can cause a lot of problems in hospitals. So, because of the growing problem of antibiotic resistance, loads of suggestions have been made to reduce this problem, reduce the prescription of antibiotics, encourage patients to complete the course of antibiotics, screen and quarantine infected patients so other people don't end up with the same infection, and then more people have to take antibiotics. And obviously follow a very strict hygiene code of practice in hospitals to prevent the spread of infection. Now, again, common things that the World Health Organization tries to promote to reduce antibiotic resistance: don't share antibiotics, never use leftover antibiotics, always take the full prescription even if you feel better. This is a really important one. Patients feel they can take it for a couple of days and then they'll feel fine. Then they're like, okay, maybe I don't need them anymore, but that's what causes the biggest problem. Take the course completely. And obviously a lot of this is infection prevention. Wash your hands, avoid contact with sick people, keep your vaccinations up to date. Now, I don't know what's happened here, but you can't seem to see the text. Bear with me 1 second. Okay. So when we talk about vaccinations, we're talking about the injection or ingestion of harmless antigens. Key point here is harmless. They doesn't really cause much problems in the human body. They are either detached from the pathogen or they're dead, weakened pathogens, okay? The whole point is to generate that immune response of antibodies. Remember, vaccines, humoral immunity, they work synonymously. It's the whole point of making those antibodies, creating those memory cells, so should we have reinfection, we know exactly what to do. And that's the whole point of the COVID vaccine as well. Immunizations is something we've been having since we were babies. I mean, your first immunization starts at 8 weeks and it goes on and on until you're an adult, okay? Loads of different vaccines for loads of different diseases. And vaccines are actually so important because they've wiped out some really, really terrible diseases such as polio, smallpox, rubella, diphtheria. And it's reduced significant deaths from things like measles, mumps, pertussis. So if you remember the MMR vaccine, measles, mumps and rubella is a very, very common vaccine, and it's the one that's had a lot of controversy around it as well. But they are really, really important in helping us fight infections that previously wiped out populations and nations. The whole point of vaccines and the whole sort of concept behind them is that if you have a population, okay, that's been vaccinated, what we have is if you look at the top example, we've only got 4 people vaccinated. We now have this many people susceptible to infection, okay? And it means that this many people can then get infected and spread the disease. Where we have a population that is very well vaccinated, we go from maybe 10 people that could potentially carry an infection to maybe 4 people that can potentially carry an infection. And the whole idea is that if many people are vaccinated, the disease does not spread. The more people that are vaccinated against COVID, the less the disease is going to spread. And that's the sort of whole concept and theory behind this robust vaccination program. Monoclonal antibodies. When we talk about monoclonal antibodies, they are antibodies that are produced in large quantities outside of the body, okay? And they have many, many uses in modern medicine. Some of you said pregnancy testing, some of you said to recognize specific antigens. They are all very, very correct. I'm going to try and, I mean, a lot of you did tell me how it's made and a lot of you were along the right line. So I'm going to try and simply put the process on a little diagram. What's basically happening is we're going to take an antigen that we require antibodies against, and we're going to infect this mouse with an antigen. Because it's a mammal, we know that naturally this mouse is going to stimulate its B cells to produce plasma cells. So a mouse is going to be exposed to the antigen or toxin, and the mouse's B cells produce a mixture of antibodies. Now, these B cells are extracted from the spleen via surgery. Now, the thing is, we want rapid production of our B cells and your antibodies, therefore. So what we do is we actually combine the plasma cells with the tumor cells. So remember, tumor cells are notoriously known for very, very rapid multiplication. So what we do is we combine the tumor cells with the plasma cells, and a chemical is added to fuse the two. And what basically happens is when we fuse these two together, we now have something called a hybridoma cell, which then are separated and cultured. So we then end up replicating the hybridoma cells. And now what we have is a cell that has great replication capacity mixed with a cell that is able to produce our specific antibody. And what then happens is each clone is tested to produce the required antibody, and then the clones are reproduced on a large scale to produce the antibody we want. So the whole concept basically here is we want to produce an antibody for a specific antigen, but we want to do it in the quickest way possible. So we take cells that produce antibodies and we take cells that reproduce quickly to produce lots and lots of antibodies for our desired antigen in a very, very quick way. Monoclonal antibodies are very, very useful. They can be used for cancer treatment. If we know what a particular cancer looks like, we can use the antibodies to deal with that cancer and remove that specific cancer cell. Monoclonal antibodies can be used for antivenoms. If we know what a particular type of antivenom looks like, when a patient is intoxicated with the venom, we can basically quickly give them the monoclonal antibodies to clear out that venom. Diagnosing diseases. Some very, very important diseases can now be diagnosed using monoclonal antibodies. It can also diagnose specific cancers, especially that of prostate cancer. So when patients have prostate cancer or are suspected to have prostate cancer, they have a specific antigen being released into the blood, and we can then use monoclonal antibodies to detect that particular antigen and basically query whether this patient is likely to have prostate cancer or not. The other one that monoclonal antibodies can be detected for is pregnancy testing. So when a patient is pregnant, they produce lots of HCG, which is called the human chorionic gonadotrophin hormone. Now, this hormone is excreted in the urine quite generously, and basically your pregnancy test kits actually have a monoclonal antibody present that is specific for the HCG. So when you test the sample, when it's binding to the HCG hormone, it ends up turning into a different color, which then indicates the positive or negative pregnancy test. So again, a very, very common everyday use of monoclonal antibodies. And the reason it works well is, like I was saying, it's the antibodies that are attached to a blue pigment. I mean, this is green. So when it binds, it then releases that dye. So the more HCG it binds, the more positive it's going to become. So there are some ethical issues around the use of monoclonal antibodies, and it's important to be able to illustrate these arguments in certain exam questions. It's the whole idea that we're using animals in science. I mean, the tumor cells are made deliberately, and you're kind of inducing cancer into mice, and that's not very nice. Although many treatments using monoclonal antibodies have been successful, there was a study a very long time ago that actually said that some deaths have been associated with their use in multiple sclerosis, which is a neurological condition. Excuse me. In 2006, 6 healthy volunteers took part in a trial of new monoclonal antibody treatment. And what actually happened was within minutes, they were all in organ failure. Not too sure entirely what happened, but this is just emphasizing that there are some risks associated with this, and it does make people a little bit more apprehensive in their use in modern medicine. We know that some advantages of using this type of treatment is that once discovered for our specific antigen, they're highly reproducible. And we know that it only binds to the diseased cell, so therefore the healthy tissue is not affected at all. So it's a very, very good way of dealing with disease. And obviously, because it's so specific to the type of tissue, and it's really, really binding to the infected tissue, there's less chances of side effects as well. It's The disadvantage, as we know, and some of the things you can mention in your exam questions, is very expensive and a lot of research is required. There are so many diseases in the world, and different monoclonal antibodies will be needed for different diseases. They sometimes can have more side effects than expected, because remember, we're initially testing the theory in mice. Mice and humans are very different. So the next thing we want to talk about is developing drugs. Now, before we take some paracetamol, before we take some ibuprofen, before we took penicillin, all of these drugs took a very, very robust drug production line in order to get onto the shelves. A new drug, it needs to be effective, safe, stable, and it must be able to meet the needs of your body. It needs to be effectively absorbed and removed from the body as well. Now, when developing new drugs, all these conditions have to be met, and the new drug must perform better than the standard treatment. Otherwise, there's no point deploying all that money into developing it. Now, the process that takes place when we're developing new drugs is actual discovery. We then have development. We then have, so basically the whole idea is that we discover, okay, there is a potential substance that can deal with disease X. We then develop a tablet out of it. We do some preclinical testing in the labs on mice, etc. Then we do clinical trials on humans. And should they be successful, we then have approval. And this can take up to 10 to 15 years. So when we talk about discovery of drugs, we often discover drugs, and they're often made and developed from plants and fungus. Some very common ones you're expected to know for your exam is basically aspirin. So aspirin is a painkiller, and it's also a tablet used to prevent coronary artery diseases, etc. And it's a painkiller developed from willow bark, which is here. So this is your aspirin. And in some of your specifications, it actually specifically identifies that this is things you need to know. Digitalis, again, digoxin is the actual drug name, comes from a plant called foxgloves. And penicillin is an antibiotic made from fungus. So there are loads of different ways in which we can discover our potential medication. So the first point is discovery. Oh, I have a plant here. Maybe this can be used to develop into a drug. So then what we do is we then conduct lots of experiments on this potential drug and tablet. And we talk about and we try to figure out how it's absorbed, how it's going to work in the body, what its potential uses are, what's the best dose, and how it interacts with the drug. So lots and lots of research happens. And we end up with one particular tablet. So then we test it on physiological environments. We can do this on cells, we can do this on tissues, we can do this on computer models, and we can do this on live animals as well. And again, we are looking at toxicity, side effects, effectivity, before we take it to the humans. So then the drugs are tested on people, and the clinical studies are designed very, very carefully, and they happen in a very, very particular way. Phase 1 is basically where we test it on healthy people. And it's a very, very small scale study because we're trying to say, is this safe and what is the best dose? Phase 2 is basically where we do it on the first intended patients. So whatever treatment we were trying to initiate, and we do it on a couple of 100 people, and we're seeing, is this effective? Does it have any side effects? And phase 3 is where we do the long-term studies where we're looking at side effects and how efficient it is over a couple of years long. Once we have passed all these stages, we can then go on to approval of the drug by different governing bodies in the UK, in the USA, in the European Union. And what you need to remember is for every 10,000 molecules screened in discovery, only one manages to pass all the stages. So remember, we discovered the drug, we are looking at a plant, and we have this massive epiphany thinking, you know what, I think this plant can deal with this condition. We then develop it into an actual tablet and test it for its pharmacokinetics, its pharmacology, how it's going to affect the body, etc. We then test it on tissues and small animals or lab models, and then we take it onto human trials. And then if it's passed everything, we go on to approve it to treat the disease we are wanting to treat it for. So to summarize, We've talked about antibodies and antibiotics. This should say antibiotics, not antibodies, that are used to treat bacterial infections. Remember, we said that antibiotics are ineffective against viruses. Antibiotic resistance is a problem in modern medicine, and steps have been taken to reduce the evolution of more antibiotic resistant strains of bacteria. Vaccines, very, very important. They have irradiated many diseases and greatly reduced death from others. And herd immunity is something we try to achieve via vaccine programs and immunization schedules. And it's basically the concept where we've got enough of the population immunized so that the pathogen can no longer spread. There are certain ethical issues associated with vaccines. We talked about monoclonal antibodies and how they're produced. So remember, antigen into a mice. Mice produces plasma cells. Plasma cells are fused with tumor cells to produce a hybridoma, and then we can harvest lots and lots of antibodies. There are many uses of monoclonal antibodies, but there are also loads and loads of ethical issues. We talked about how drugs are developed in the lab and then they're trialed on people, and we then sort of go on to put them on our shelves. So time for some exam, just sort of filtering through how many exam questions we have. Okay, good. Just keeping an eye on time here as well. So part 1D, white blood cells are part of the immune system. Use the correct answer from the box to complete each sentence. When we are ill, white cells produce what? We know they don't produce antibodies, they're not going to produce pathogens, and they don't certainly produce vaccines. They produce antibodies. Many strains of bacteria, including MRSA, have developed resistant to drugs called antibiotics. So remember, B cells produce your antibodies, and it's your antibiotics that are going to deal with bacterial infections. So what we have here, it says pathogens are microorganisms that cause infectious disease. The graph shows the percentage of children under 5 years old who died from infectious diseases in the UK in 4 different years. So we've got 1750, 1850, 1950 and 2015. Between 1750 and 1850, vaccines were also developed. What is a vaccine? It's large amounts of a dead pathogen, no. It's large amounts of a live pathogen, no, because we don't want to make the patient more sick. It's small amounts of a dead pathogen. Now it says the advances in medicine has had an effect on the death rate. Describe the effect these advances had between 1750 and 1850. So what's basically happened is between 1750 and 1850, we've had this massive decrease. So almost from 70% to 10% of death occurring in children from infectious diseases. Okay. And we can see that the effect is that it's reduced death, and then you go on to include data. So you mention the 70% to the 10%. Okay, so nice and easy. You establish that it's a decrease, and you're establishing the percentage. So again, I'm getting the 70% from here, and I'm getting my 10% from here, and this is where they get the 60 from. Nice and easy. Antibiotics were developed in the 1940s. Which one of the following is an antibiotic? Again, it's important to know your antibiotics. Now it says the use of antibiotics has not reduced the death rate due to all diseases to 0. Suggest 2 reasons why. First of all, antibiotics are not designed to deal with all diseases. They only deal with bacterial infections. Okay, antibiotics, we've got resistance happening as well. New diseases come to be, so we don't have enough antibiotics to be dealing with all of them, and it takes time for antibiotics to develop, and sometimes we can't keep up with the rate of new disease development and the rate of production of new antibiotics. So some of the things you can say: antibiotics only kill bacteria, they do not kill viruses, some bacteria are resistant, and allow things like deficiency diseases are not caused by bacteria, inherited diseases are not caused by bacteria, lifestyle diseases are not caused by bacteria. So the key thing here is in your exams, all diseases. Okay, this is what they're referring to. And remember, we said that if we talked about so many different diseases, we talked about non-communicable and we talked about communicable diseases. And it was the idea that even the non-communicable genetics, the deficiencies, the cancers, you can't deal with antibiotics. The communicable diseases caused by bacteria, viruses, fungus, and parasites, again, antibiotics are only going to be useful for the bacteria one, not these. So you can't use them to eradicate all disease. Now, in school laboratories, bacteria should be grown at a maximum temperature of 25 degrees Celsius. Give one reason why companies testing new antibiotics grow bacteria at 37 degrees Celsius. Now, basically, one of the reasons why we grow bacteria at 37 degrees is because it replicates the human body temperature and it's the rate at which pathogens grow. Okay, now obviously in a school laboratory, we're not going to try and grow masses of bacteria, but obviously in drug testing companies, we want to be able to grow it and we want to mimic the growth rate of the body. Question 7. A virus called RSV causes severe respiratory disease. Suggest 2 precautions that a person with RSV can take to reduce the spread of the virus. Remember, catch it, bin it, kill it. Wash your hands, wash your surfaces, stay away from people if you're infected, cover your mouth when you're sneezing, wash your infected clothes. So all these sensible suggestions will get you the marks. So it says 1 treatment RSV uses monoclonal antibodies that can be injected into the patient. So it says the scientists can produce monoclonal antibodies using mice, and the first step is to inject the virus into the mouse. So then it says remaining steps to produce the monoclonal antibodies. So remember, we inject the mouse produces your antibodies via its plasma cells. Plasma cells are infused with a cancer cell to produce a hybridoma, which then goes on to produce our desired antibody. So nice and easy explanation. So again, hand washing, sanitizer, cover nose, mouth, put used tissues in the bin, don't kiss uninfected people, fair point. And then when we talk about this, and I think this question is a very nice, simple way to learn about monoclonal antibodies. If everything I said earlier confuses you, just take this exam answer as a revision for what your monoclonal antibodies are. So remember, we stimulate the mouse's lymphocytes to produce antibodies. We combine the mouse lymphocyte with a tumor cell, and it creates a hybridoma. And we clone the hybridoma cells, and they rapidly divide to reproduce the antibodies. So anything along these lines will suffice. Now it says, describe how injecting a monoclonal antibody for RSV helps to treat a patient suffering from the disease. So basically, what's going to happen is the monoclonal antibody will then go on to bind to the RSV virus, stop it from further infecting the patient. Okay, and when the monoclonal antibody binds, it allows the white blood cells to then go and engulf the virus. Okay, so it's the whole idea that the monoclonal antibody is going to be complementary and it will bind to the virus, and this will initiate the removal of the virus from the body. Now it says a trial was carried out to assess the effectiveness of using monoclonal antibodies to treat patients with RSV. Some patients were given a placebo. Remember, the whole purpose of a placebo is we need a control, because we want to see if we were giving them nothing, what kind of response we would have, and the control is often used to compare the effects of treatment to no treatment whatsoever. Now it says a number of patients had to be admitted to hospital as they became very ill with RSV, and the results are shown in the table below. So we've got treatment received by the patient is group A, monoclonal antibody, and the number of patients within each group admitted to hospital with RSV. And then it says a group B, which is the placebo. And already we can see the ones who were given the placebo and had RSV, a lot more of them were in the hospital. Now it says the trial involved 1,500 patients. Half of the patients were given the monoclonal antibodies in group A, and half of the patients were given monoclonal antibodies and half of the patients group B were given the placebo. Okay, so it says calculate the total number of patients admitted to hospital with RSV during the trial. So if we have 1,500 patients in total, first of all, we're going to take 10.4 and 4.8, okay, which is the percentage of patients within each group. We're going to divide it by 2, and then we're going to further divide it by 1,500 to find the total number of patients. So when we have a look at it over here, what we've basically done is we've taken the 4.8 and the 10.4, okay, and we've found the total percentage of patients. Okay, we've then divided by 2 because only half of these patients were given the drug, and then we've divided by 100 to get a percentage, and that value has then been multiplied by 1,500 to calculate the total number of patients admitted to hospital. So what we've basically done is we found these values of 1,500. Okay, that's all. So again, it's a little bit of, I'm not going to say heavy math, but it can get really, really confusing. So just take a minute to look at the numbers in front of you and look at the information they've provided. Then the last part of this question says, evaluate how well the data in the table supports the conclusion monoclonal antibodies are more effective at treating RSV than a placebo. Now, to a certain extent, this is correct because the percentage of patients who went into hospital while being treated with a monoclonal antibody was significantly less. Okay, when we compare it, we're looking at 4.8% patients in comparison to 10.4. However, that doesn't go on to say that no patients were admitted when they were on the monoclonal antibody treatment. So these are the sort of points you have to make. So as you can say, it supports the conclusion because over the double number of patients in the trial were hospitalized with the placebo. Should any of you have gone for the other side of the argument, as long as you have presented your point well, you can get a mark. So you can say things like, the data does not support the conclusion because some of the patients, there's no information on patients who were not hospitalized. What if they were ill at home? What other factors may have affected those patients who were in hospital? Okay, so then you can pick it apart like that. So I believe that takes us on to the end of, oh, not yet. Okay, never mind, I take that back. So question number 8. Many people in the UK take sleeping pills. The drug thalidomide was developed as a sleeping pill in the 1950s. In the 1960s, thalidomide was banned. Recently, thalidomide is now used to treat other diseases. Now, I actually mentioned this in a revision session I did, but for those of you who do not know, thalidomide is now used to treat things like leprosy and bone and blood cancers. Again, a type of general knowledge that you just have to accept and know. Now it says the table here shows information about the development of a new sleeping pill. And what we've basically got is we've got preclinical, clinical phase 1, clinical phase 2 and clinical phase 3 and what it's being tested on. What is the shortest time taken to develop a new sleeping pill? As we can see over here, we just need to figure out the different years. So here we've got 1 to 4 years, which is a total of 3 years. We've got 2 to 4 years, 1 to 3 years or 2 to 4 years, and we can say it's a total of 6 years that it's taken us to develop that pill. Okay. Now it says, what is the range for the number of volunteers needed to complete all the clinical trials for the new sleeping pill? Now, can you see over here, we've got a range of basically 20 to 5,000 volunteer patients. Okay. Now, what we can basically sort of derive from this is you need to sort of have a look. So you need to sort of have a look. Let me just get rid of this. You need to sort of have a look at what the range they've given you and sort of be able to elaborate on a figure from that. Okay. So and they basically given us a value from 11,020 to 5,600. Okay. So again, it's about looking at what the data they've given you to be able to derive that. And what I was emphasizing when we were sort of covering the whole concept of pills and how we're developing them, what you can sort of see is, remember in the preclinical trials, loads and loads of compounds are being tested. And by the time they've been trialed and tested, we now basically go on to looking at around only 5 to 10 compounds that are being tested on a handful of volunteers. And out of the 5 to 10 compounds, only 2 or 3 are actually going to come through and be tested on patient volunteers. And even from that, only 1 new sleeping pill is going to be produced. One of the reasons why we have such robust testing systems now is because we have issues like things with thalidomide. For those of you who know, thalidomide was initially used as a sleeping pill, but had to be taken off the market because pregnancy women were taking it to treat morning sickness, and it led to loads of birth deformities in children, and it was taken off the market. So now anything that's ever redeveloped has to go through a robust system of testing and before it can be officially approved. So then it says drugs are trialed to check for side effects on people. Give one of the reasons why drugs are trialed. We want to know how effective this drug is. We want to know how it's going to interact in the body with other drugs that the patient may be taking. We want to know what the most effective dosing is for the patient. We want to know whether there's any efficacy. So remember, they've already said you check for side effects. So saying things like allergies, side effects, and safety is not going to give you the marks. You want to say things like, we're testing for toxicity, we're testing for ideal dosing, and we're testing for efficacy. Okay. So the last part of this question basically says, the pie chart shows the impact on the population caused by drugs from different sources. And we can see we've got 3 different types of sources here: legally prescribed drugs, legal non-prescribed drugs, and illegal drugs. Legal non-prescribed drugs have a greater impact on the health of a population. Why? Well, remember, if they're legal non-prescribed drugs, now the thing is, they can be easily abused. Now, even though they're legal, there is no sort of, so remember on prescription, you have to go to the doctors to renew your prescription every single time. If they're non-prescribed and you're taking them over the counter, there's no regulation of how many you are taking and how many you're having. And that can lead to development of drug addictive habits and drug abuse as well. Okay, then it says drugs change chemical processes in a person's body. Why is it difficult for a person to stop taking certain drugs? Because they're addictive, they become dependent on that particular drug. So again, it says, if you look at some of the information here, it says more people take or use legal drugs, and legal drugs are more readily available, and these can become more likely to be abused. Okay, so common legal drugs: alcohol, tobacco, and they use more, they have more devastating effects on the body as well. So again, when we're talking about the reason why patients can't stop taking drugs, it's the level of dependence and it's the level of addiction. And often, a lot of the time, stopping the drug isn't the hard thing, but it's the actual dealing with the withdrawal symptoms. If you attempt stopping, that becomes difficult. If any of you guys are interested, you should take a look at what happens when a patient stops and abstains from taking alcohol. The things their body goes through over the next 72 hours really will explain to you why it's so hard for some patients who are addicted to stop. Okay, so that takes us on to the end of the conversation about modern medicine. Again, I feel like I've thrown a lot of information at you, but I hope this information you can use to effectively answer your exam questions. The exam questions a lot of the time are data interpretation when we're looking at clinical trials, or they're basically straightforwardly asking you, how are monoclonal antibodies made, how are vaccines made, how are they useful? So you should be able to be comfortable with these concepts now. This then takes us on to the last part of the session, but before we move on, I just want to ask you guys, just to make sure someone is there on the other end of the screen as well. How confident are you with this topic now? So the topic I'm specifically referring to is the use of monoclonal antibodies. Oh, we have 100% coming in. Monoclonal antibodies. Are you able to define the use of antibiotics? Are you able to establish the use of antibiotics? Are you able to say vaccines, what they are and what they're used for and what type of immunity they help develop in society? Can you explain how a drug is made? So we have a couple of mixed responses, and take this time to be able to evaluate what particular thing you are slightly confused about, whether it's the actual content itself or whether it's applying the content you've learned into the exam questions. And that's that's basically then going to be you deciding how you need to tailor your revision to when you can tailor your revision to when you're really looking at how to maximize your marks. Okay, good. So like I was saying before, we are now going to move on to the last part of the revision session. I am very well aware that we have the better half of 20 minutes left, but I promise you I don't have a lot more to say. The next part of the topic is actually quite nice. We're talking about some light biology to do with plants. So let's have a look. We're going to be looking at plant diseases, and we are going to basically look at 3 common diseases in plants. So I'm going to touch on the tobacco mosaic virus and the rose black spot virus in plants. We're going to look at the different types of plant infections caused by your viruses, bacteria, and fungi. We also want to detect how iron deficiencies can affect plant growth. Okay, and we want to look at some of the ways in which plants can defend against animals, how plants produce chemicals that help defend them against predators, and what the physical defenses of a plant are like as well. So again, refer to your specific exam board. Now, my first question to you guys is, before I start telling you, tobacco mosaic disease is caused by which type of pathogen? So tobacco mosaic disease is caused by which particular pathogen? Again, I feel like just saying it on autopilot should suggest what it's being caused by. What does it look like in plants? How do we avoid the spread of this particular disease? So the tobacco mosaic virus is a disease caused by tobacco mosaic virus, and it affects plants. Rose black spot is caused by which pathogen? A virus, fungus, or bacteria? So the rose black spot is actually caused by a fungus. Contrary to popular belief, I sometimes associate it with a bacterial infection, but it's actually a fungal infection. Plants use nitrates to convert which component into what? Do they use nitrates to convert sugar to protein, proteins to sugars, water to carbon dioxide, or carbon dioxide to oxygen? So plants will actually use nitrates to create protein. So what they end up doing is they use the sugar and to translate it into protein they can then use for growth and plant processes. Chlorophyll is made from which ions? Which ions do plants have to use to make chlorophyll? Is it calcium? Is it magnesium? Is it sodium? Is it nitrates? Okay, so magnesium is the correct answer. Plants actually use magnesium to generate chlorophyll, which is the pigment they need to generate photosynthesis to create glucose. So let's have a look at some of the plant diseases. Now, the tobacco mosaic virus, as just explained, is a virus that causes diseases in many plants. And basically, one of the common plants it causes disease in is your tomatoes. And the symptoms basically are that you have this mosaic-like discoloration and damage on the leaves. And the thing is, it's actually very, very easily spread. So we have to get rid of the entire crop before it affects the entire produce. Okay. Now, rose black spot is a fungal infection that causes disease in roses. And as you can see over here, you've got these purple black spots and yellow leaves that then effectively fall off. It prevents photosynthesis and reduces the growth of these plants. And again, you can treat it with fungicides. And again, you can't really give these antibiotics and fungicides, so you literally just have to get rid of all your infected plant crop. Now, certain minerals are used for plant growth, and certain minerals can result in deficiencies in the plant if we don't have enough of them. Plants need minerals. Some insects, such as nematode worms, can actually damage plant roots. And what that basically means is once the plant roots are damaged, so remember the plants often get all their nutrition from their roots from the soil, it prevents the plants from getting minerals and water. Now, one of the key minerals the plant needs is nitrates. As you guys previously identified, nitrates are actually used to convert sugars into proteins. And where we have a deficiency of nitrates, the plant cannot grow properly and its growth becomes stunned and it basically just stops. And you can see over here in this experiment here, we have a plant that's got plentiful nitrates and we have a plant that doesn't have nitrates. It's dull, it's not so green in color, and it's just not flourishing as well as this one is. When we have a lack of magnesium ions, your plant becomes very, very pale. As I was showing you before through the revision questions, magnesium ions are actually used to make chlorophyll. And chlorophyll is where photosynthesis takes place. So where you have plants that are very, very deficient in magnesium ions, they lack chlorophyll, so they don't photosynthesize. So the plants won't have this rich green color and they will turn yellow and growth will stop. So minerals are only important in humans. They're very, very important for plant growth as well. Now, there are many different ways in which we can detect disease in plants and basically identify that this plant has something going wrong with it. So if a plant isn't growing well, we can start thinking about the fact that it's got a lack of nitrates. If there are spots on the leaves, we can start thinking about using antifungals because they may have the black spot fungus growing. If they have decay, we can start thinking about, we need to get rid of this because they're decaying. So this is an example of potato blights because our potato crop has decayed. I mean, it looks awful. And growth, if the plant ends up growing really, really weird calluses, like things like this, we can start saying, okay, you're being infected by a bacterial infection. I need to do something about it straight away. And obviously malformations of stems and leaves can be caused by pest infections. And we have already identified that discoloration can be caused by things like magnesium deficiencies, or sometimes it can be indicative of the tobacco mosaic viral patterns. obviously there are certain pests that can be present on a plant as well, like little aphids, and you can see them here. I've done a very clear picture for you guys here, and they can be very, very visible and they can completely destroy a plant. So loads of things you can do to detect disease on your plant. Now, obviously, like humans, plants also have defense mechanisms in place. They have physical barriers that can reduce the number of pathogens entering the plant. They have cellulose cell walls that are very hard and strong and prevent penetration from pathogens. A waxy cuticle, again, another physical barrier. Bark, so trees are full of bark. Their entire trunk is bark, and it makes it very, very hard to penetrate and be eaten and ingested by predators and pathogens. Loads of layers of dead cells on the stems actually make it a very, very hard layer again to penetrate through. And leaf fall, often magically, and just the way nature is, infected leaves can naturally fall off, and this is a very, very protective mechanism for plants because then it prevents the infection from spreading to other parts of the plant. There are chemical defense mechanisms as well. Plants actually have lots of antibacterial chemicals that prevent bacterial infections. Plants actually also protect themselves from being eaten by animals. They're not just protecting themselves against pathogens. Some of the defense include things like poisons, thorns that make it painful for the plant to be eaten, such as things like that on a cactus. And these all work to sort of deter the animal away. Hairy stems and leaves basically prevent insects and large animals from feeding on them and laying eggs on them, so that it makes it a very hostile environment. And leaf movement, and often some plants can droop or curl when you touch them. This is very, very rare, but it is a very, very effective adaptation, and it dislodges insects and scares large animals away. So again, plants aren't just defenseless, they've got physical barriers, chemical barriers, and defense mechanisms in place as well. So again, to summarize, plants can also be affected by pathogens. We've talked about the tobacco mosaic virus and the rose black spot fungus. And they can suffer from mineral deficiencies. Lack of nitrates leads to stunted growth. Lack of magnesium leads to lack of chlorophyll, which leads to very, very pale, unphotosynthesizing plants. You can observe your plants to see if there are any changes and signs for disease. We know that plants have physical and chemical defense mechanisms to protect them against certain pathogens and herbivores. Now, having a look at some of the exam questions, I think we have just 2 exam questions to do, and I think it's a very nice way of sort of summarizing some of the ways in which they can ask you about this particular topic. The tobacco mosaic virus is a disease affecting plants. The diagram below shows a leaf infected with tobacco mosaic virus. We have yellow patches here where the tobacco mosaic virus has destroyed the chloroplast. All tools should be washed in disinfectant after using them on a plant with TMV. Why? Well, obviously you want to reduce the spread of it to other leaves. Scientists suggest producing a single plant that contains a TMV resistant gene. Suggest how scientists can use this plant to produce many plants with the TMV resistant gene. So you basically can use a cloning, so different types of tissue cloning methods can be used. Okay, so you take stem cells from the meristem or tissue culture. So you either grow the plant or you take the tissue and replicate that tissue to produce the same type of plant again. Now it says some plants produce fruits which contain glucose. Describe how you would test for the presence of glucose in fruit. Now again, I thought this was a very, very random question to ask, but what you do is you actually use the Benedict's test, and I'm sure I actually did this in high school, so I'm sure you guys must have too at some point. Basically, when Benedict's solution is actually initially blue, when we have glucose present in it, it turns to orange. Again, just a standard common experiment done in biology to test for the presence of sugar. Now it says TMV can cause plants to produce less chlorophyll. This causes leaf discoloration. Now explain why plants with TMV have stunted growth. If we're producing less, hang on. So if we are producing we have less photosynthesis, less production of sugars, and less means of growth. Okay, you can't convert the sugars into proteins and less growth. Okay, it all comes back to to lack of photosynthesis. So again, you can say things like less photosynthesis because of lack of chlorophyll, less glucose made, less amino acids or proteins, so therefore reduced growth. Now, rose black spots is a disease of the roses. Remember, I already established that this was a disease caused by a fungus, not a bacterium. The virus you need to know about is the TMV virus. Now, unfortunately, the answers have already come up on this, but it says here, explain how different types of organisms defend themselves against microorganisms. So what you need to basically be able to do is, if I go to, you can see that this is actually worth 6 marks, okay? And it's quite important to be able to establish where you're going to get those 6 marks from. First of all, it's the fact they're asking about different types of organisms. So we want to talk about my pen doesn't seem to be working. So we want to talk about both animals. Okay, let's try that again. So, okay, fabulous. So we want to be able to talk about animals, plants, and fungus. Some of the recommendations they've made are things like animals defend themselves with their skin, their nose, their stomach, their white blood cells. So all the defense mechanisms we talked about earlier on in the day, that's your animals. We've talked about the plants. We've said they've got the cell wall, the waxy cuticle, the thorns, and you know, the fact that they can produce antibacterial chemicals as well that prevents other people from attacking it. Fungi can actually produce antibiotics which kill bacteria. So again, remember, bacteria is a pathogen, and we've previously said that we can use fungus to produce antibiotics to kill bacteria. So when they talk about other organisms, you need to be able to mention all these kingdoms, the animals, plants, and fungus. Here we have an experiment where students have tried to grow some bacteria in the laboratory. They've basically used a inoculating loop where they've then taken the bacteria from and put it onto a petri dish. It says they've removed the lid of the petri dish, they removed the lid of the bottle containing the bacteria, they use the inoculating loop to remove some of the bacteria, and then they spread the bacteria over the agar. They put the lid back on and they put the petri dish into an incubator. Now it says give 3 improvements to the method to prevent contamination. So I think, first of all, they're removing the lids of everything. I think they should keep the lid closed for the petri dish and then open this one, then close that one and then open this one. Okay, then you can talk about things like making sure you're only using the one inoculating loop. You can talk about maybe the temperatures at what they're being put at as well. So there's various things you can talk about. So remember, talk about sterilization. So this was the idea of keeping the lid on to make sure you're minimizing exposure of agar to the air. You can sterilize your agar dish, you can sterilize the petri dish, you can even sterilize the inoculating loop. I think this is what we used to do in our classes, where we would secure the lid of the petri dish with tape to make sure nothing could get in. Now it says, why did the student grow the bacteria at 25 degrees then 40 degrees? Because we are trying to prevent the growth of harmful pathogens. And I believe that takes us to the end of this session. So again, are you comfortable with the different types of diseases that affect plants? Are you able to establish which ions cause which deficiencies in plants, and how do plants protect themselves? Wow. So I think you'll be very glad to hear me say we are now officially at the end of the revision session, teaching session, wherever you want to sort of put it as. We have covered an awful lot of topics. We have covered a lot of content, and I want to say a big well done to sort of staying on and really pushing through it. I really hope this session has been useful to you guys. Do fill in the feedback form. It's very, very handy for us to sort of see how we can improve the lessons, to sort of improve your experience of it. Again, to summarize, we've talked about what a pathogen is. We've talked about the different types of pathogens there are. We've talked about how the body's innate immune system and adaptive immune system deals with pathogens. We talked about drug development. We talked about monoclonal antibodies and what they're used for. And we've also talked about pathogens and disease in plants. Again, a lot of things to cover on a Saturday morning and afternoon. Very well done for your interaction. I hope you found this session helpful. Again, do leave your feedback in the feedback form, and I will see some of you guys tomorrow. So I can see 1 or 2 questions coming in through the chat that I am tempted to answer. I think there is a question about how you would seal the Petri dish. You just put some adhesive tape around the sides and hope nothing's going to contaminate your dish. I use a Surface pen on a Surface Pro tablet. So again, please, if you're still around, don't forget to fill out the feedback form, and we're sort of going to call it a day now. I think it's been a very, very heavy session, and I will see you guys tomorrow.